New South Wales DPI Tocal College. Welcome to the 44th Annual Tocal Beekeepers Field Day. Online once again, but we hope to return to a great face-to-face event next year and catch up with new and familiar faces after just on 12 months of management of Varroa in New South Wales. Always nice to have something to look forward to. Firstly, I would like to acknowledge that I am hosting the webinar from the lands of the Wanneroo people. I also acknowledge the traditional custodians of the various lands on which you're participating from and any Aboriginal and Torres Strait Islander people with us today. I pay my respects to elders past, present and emerging and celebrate the diversity of Aboriginal peoples and their ongoing cultures and connections to the lands and waters of New South Wales. It's hard to believe that just on 16 months ago, Varroa destructor was detected at the Port of Newcastle. So much has taken place in this short period of time in our beekeeping history. A huge response effort has been undertaken to contain and eliminate this major pest of beekeeping globally. And although this path has not been successful, all respect must be given to those who sacrificed so much. I'd now like to introduce our first presenter, Education Officer.
Welcome to Tocal College Beekeepers Field Day. I'm Kelly Lees, Education Officer Honeybees for the New South Wales DPI and Tocal College. And today I want to talk to you about Varroa mite. Varroa is a parasitic mite of the genus Apis, or honeybees, which includes Asian honeybees and European honeybees. Originally, it was a parasite of Apis cerana, the Asian honeybee. Now, Varroa and Apis cerana have had hundreds of thousands of years to build up a stable host-parasite relationship. It's had Varroa for a long time. So Asian honeybees have developed a suite of behaviours to cope with having Varroa in their colonies, including absconding and swarming. But these behaviours make them unsuitable for the style of beekeeping that we use in the West, which is why we have the European honeybee, which is more amenable to being kept in hives. Now, we now have Varroa on all inhabited continents. Australia was the last remaining continent until it was detected in June 2022, and Varroa has now been declared endemic in New South Wales. It's here to stay, and now we have to live with it. European honeybees haven't had Varroa for very long, less than 50 years. So they haven't had a long time to build up a host of behaviours like Asian honeybees have to cope with Varroa. So it causes quite heavy impacts on European honeybees. They're just not ready to deal with it as well as Asian honeybees yet. So it's a very recent jump across to European honeybees. How does Varroa work within a hive? Well, it only takes one foundress mite. So a bee returning to a colony will bring back a Varroa mite, often hidden between the abdominal segments and often underneath, so you don't see it come in. That foundress mite, once she's inside the hive, jumps off that adult bee and finds an uncapped brood cell. Now, Varroa have preference for drone brood cells because they're capped for a bit longer and they're just a bigger larvae to feed on. However, a colony that has a lot of worker brood will compete with the mites for the drone brood. So you'll always have a big mite load if you've got a strong colony because there's lots of brood cells for the Varroa to choose from. So that mite will go into that open brood cell. She will go down and burrow into the brood food at the bottom of the cell and she'll hide until that cell is capped. Once that cell is capped, the Varroa mite will emerge and she will start laying eggs. She will also commence feeding on that larvae, so injuring that larvae, feeding on the fat body in the larvae. Those eggs will begin hatching. The first few eggs will be male, followed by female mites. They all start feeding on the developing larvae, causing lots of health impacts for that poor bee. Once they're mature enough to reproduce, mating occurs in that capped brood cell. Then once that adult bee is ready to emerge, the mature female mites will emerge alongside it. The immature mites and the males remain in the cell and actually die, but the mites that have reached maturity and mated will go out and they'll find another empty cell in which to reproduce in. Before they do that though, they do jump onto the adult nurse bees and feed for a period of time before they go back into a brood cell in order to complete and continue that reproductive cycle. What are the impacts of varroa mite on honeybees? You've got impacts on individual bees and then there are impacts on the colony. So for an individual bee that's been fed on via varroa in a cell before it emerges as an adult, when it does emerge, it weighs less than its counterparts who haven't been fed on. For worker bees, that's around 7%. For drones, it's even higher, between 11 and 19% less weight than a drone that hasn't been fed on via varroa. So they're significantly smaller. That impacts on their lifespan, so they don't live as long as other bees that haven't been fed on. There are impacts on their cognition. So cognition, the way they think. So they go out, impacts on their flight and navigation. They sometimes don't return, so you get long absences from hives. They get lost or they return without food. So they can't navigate properly and they do poor foraging. There's also an impact for worker bees on their hypopharyngeal gland. That gland is very important for brood feeding. So they can't as effectively produce brood food for the young larvae. So not only are those new larvae being impacted by varroa, but they're not getting as good quality food from the nurse bees. There's also an impact on the bees' immune function, so they're more open to getting viruses and other diseases. There's also for the colony a breakdown in social cohesion. So because their cognition and thinking ability is affected, they also don't groom each other as much. They don't groom themselves as much. They also don't share food and nectar, so that trophallaxis, that reduces. So there's just a general breakdown in the continuity of how the bees communicate within the hive, which affects the colony. You can also get that leading to what's known as parasitic mite syndrome, where social cohesion is completely lost, other diseases take over, and you get lots of different impacts on the health of the colony. Now, we don't know about PMS in Australia yet because the varroa that we have luckily doesn't have any viruses. And usually parasitic mite syndrome is associated with viruses. So hopefully we don't start to see that because that's implicated in colony collapse. How does varroa spread? Well, it spreads naturally over time. Generally, natural spread is quite slow. Bees from colonies have to encounter other bees from infected colonies in order to have a varroa jump ship and head back to its colony. So that spread just creeps along very slowly. There are other ways that it spreads though, and the main way that it's spread is by beekeepers. So an infected colony, you get drift, so bees in an apiary or a load, you've got one infected hive and bees will drift within an apiary. And so a bee that's infected or a colony that's infected will drift bees into another hive, so you can get natural drift. You can also get robbing. So say a feral colony has been invaded by varroa, it's weak, it's got other diseases, you've got a beehive, it's sensed a weakness, it's gone out and robbed a colony that's got varroa mite. Those mites then hitch a ride on your bees back to your hive, and then your hive is infected. And then they can encounter them when they're foraging. So when they're out foraging, sometimes a varroa can jump ship on another bee and get taken back to a hive. But beekeepers need to be mindful that they can infect other hives by moving equipment, so sharing frames, sharing boxes, interchanging all of that equipment can move varroa from one hive to another. So it's really important that you're actively monitoring for varroa in your apiaries. Training and education in this space that we're in, and it's a new space, is absolutely critical. One thing you need to carry on doing is conducting surveillance in the varroa mite management zone. If you're going to get bees again, you need to be thinking about monitoring, not thinking about doing. You need to be monitoring for varroa mite in your hives every 16 weeks and reporting those results. So if you find varroa, letting DPI know. If you're in the varroa mite suppression zone, so the green zone outside of those orange zones, you still need to be doing your monitoring as well. You are monitoring for mite arrival. We need to know the natural spread of the mite and how far it's gone. So keep up with your surveillance and monitoring, absolutely critical. And then there's treatment. Obviously we're not treating yet, but at some point we will be. There's lots of treatment options, and I'm not going to go through them all here, but there are chemical treatment options, there are mechanical treatment options like drone trapping and splitting. There are cultural methods, so breeding for varroa resistance, those sorts of things. There's lots of things you can do, but it's important that whatever treatment you do, it's done as part of integrated pest management. So a variety of treatments to help control varroa. Training and education around all of these things, monitoring and treatment and integrated pest management, will be coming. So don't worry, there is education on the way. What's really, really critical is when you source information, that you source that information from a reputable source. There's lots of things out there on the internet, and there are also lots of treatments that are done in other countries overseas that may or may not be approved for use in Australia. So don't think that you should be applying treatments to hives prophylactically or anything like that. It's really important that you get your information from a reliable. Right now, the source of for monitoring treatment is deep. So please make sure that you're not seeking information from sources that are unreliable. For more information about varroa and other pests and diseases of honeybees, please visit New South Wales DPI or the Tocal College website. On behalf of Tocal College, thank you.
I'm now pleased to introduce Madeline Kratz, Honeybee Industry Development Officer.
Hi everyone, and welcome to today's Beekeeping Field Day. My name is Madeline Kratz and I work with the Department of Primary Industries based at Tocal College as the Honeybee Industry Development Officer. And today I would like to take the opportunity to talk a little bit about nutrition for bees and for varroa mites. So the sorts of topics that we'll be covering today is, what do varroa mites actually eat? Where are mites found on bees, and most importantly also why? The capability of fat bees and skinny bees, and generally what are the nutritional requirements of honeybees, and what do we know about nutritional management of bees and the impacts of varroa. So I want to start with talking a little bit about what do varroa mites actually eat. So back in the days, or in early research I should say, people have believed that varroa mites actually feed on hemolymph, which is, you can think of it kind of like an equivalent to mammalian blood, so kind of like our blood. But since then, and that was a research done by Ramsey et al. In 2018, it was discovered that in fact varroa mites do not feed on hemolymph, but in fact feed on fat body instead. And the interesting thing is that when you look at the actual nutritional content of insect blood compared to vertebrate blood, it also kind of becomes obvious why that might be the case. So if we look, for example, at insect hemolymph, we see that less than 2% of hemolymph in fact is made up of cell content by volume. So that is in fact a very dilute nutrient profile. Whereas if you compare the cell content of insect blood or hemolymph to vertebrate blood, you can see that it actually has a much higher nutrient content, so about 40% cell content by volume, which is a much higher nutrient content. So something like, for example, comparably, if you think of a tick, for example, feeding on a mammal, they would get a much more nutrition from mammal blood compared to what you might find in insect blood. But that is a little bit of a side note. So let's focus a little bit more on the actual fat bodies that the varroa might feed on. Now, fat bodies are quite interesting for many reasons, but you can think of it in comparison to a human body as the liver. So it carries out functions for bees such as regulating water. It is also involved in detoxification of, for example, pesticides. It plays a very, very crucial role in the immune response of honeybees, but is also involved in hormone regulation and in protein synthesis. So varroa mites do get a lot more nutrition from feeding on fat body tissue. And in the process, they also transfer viruses, and that's where varroa can have a very significant impact on the health of honeybees and the colony. So where are those fat bodies found? The fat bodies are found mostly, or they are found in the abdomen of honeybees, but mostly in nurse bees and winter bees. So they are the two types of bees that are adult bees that have very high fat body stores. And there is also a reason for that, because nurse bees, they are the bees that feed all colony members. So they are the ones that need to be able to process a lot of protein, a lot of nutrients to be able to simply feed the next generation of bees. The winter bees need to have that extra storage of nutrients to be able to survive for that longer period throughout winter and have some nutrient reserves to be able to make it into spring. So in this image you can see an example of on the left of a forager bee, and on the right of a overwintering bee or a nurse bee, and you can really see very visually how the fat bodies on the right side of the image are much more developed than for forager bees. There's also fat bodies are also quite or can provide a nutrient source for varroa when they feed on immature bees, when they have entered the cell just before it's being capped, and then you have the developing larvae and the next generation of bees hatching from those cells. So this is when the varroa can also be feeding off those fat bodies of immature or developing honeybees. There's one other thing I would like to point out as we go through this talk, because it will become important later, is that you can also think of this image of fat bodies from comparing it, in this case, foragers to winter or nurse bees, but you can also think of it as well-fed bees on the right-hand side. So this is what you would be expecting compared to undernourished bees on the left-hand side, or skinny and fat bees if you like. And we will talk a little bit more about their capabilities in a moment. Now, first I would like to come back to varroa. So where do we find mites and why? Now, thinking if I was a varroa mite, I would like to be as close to my food source as possible. So I would like to ask you, what do you think is actually wrong in this image? Where is this mite located? And I think this is sort of a little bit of a misleading idea, because most of the time any articles, books, images we've seen, quite often the varroa mite is actually displayed as in hitching a ride or sitting on the back of the thorax of honeybees. And in fact, if you're thinking about looking at a honeybee colony, it will be very, very rarely that we would ever, it's basically almost impossible that we would ever be spotting a mite on the thorax of a bee just like that. So there is, in fact, people have studied how likely it is that we would find varroa mites in different parts of the body of honeybees, and there is actually only just about 1% chance that you would ever find the mite on the thorax at the back of the bee. So where are they found? So now we are looking at the underside of the bee. So it's the abdomen, but from underneath. And the varroa mite, so you can see just in the top part, is nicely tucked into the abdomen right there. And there is in fact a 20% chance that you would find varroa mite on the underside of the bees on the right hand side, tucked into the sclerite, just on the third position, but I will mention a little bit more about that later. So there's a much higher chance that varroa is found on the underside on the right hand side. But what about the left hand side? So in fact there's even a greater chance that up to 60% that you will find varroa located on the left hand side. Now you might be wondering, why is there a difference between left and right? But just bear with me. Just quickly, there is varroa mites, of course, can be crawling on bees at other body parts of the bee at other points in time, and there is about a 4 to 6% chance that you might be finding varroa mite crawling on the bee, or sort of trying to get to the abdomen, to fat bodies, potentially on different parts, but those chances are still very low, as you can see right there, 4 to 6%. Now, I hope that you seriously will never see bees covered in mites like this, because at that point in time, you can imagine that the ratio of mites to bees is much higher than you have. In other words, you have much more mites in a colony than potentially bees already. So I hope you will never ever see this many mites on the bee. Definitely is way above threshold levels, and that colony is in serious trouble. But what I would like to have a quick look at is, why is that difference between 20 to 60% on the bees, and why is it actually more likely that varroa mite are found on the underside of the abdomen of honeybees? And this is why it makes it so hard to be able to visually see varroa mites in a colony on live bees. So researchers have found that there is in fact a larger deposit of fat body tissue on the underside of the bee's abdomen. And they found that varroa mite likes to sort of tuck itself into the third segment of honeybees, and that is because it provides the best access. And in terms of being a larger pocket, I guess you can imagine it a little bit, but it also at the same time protects mites from grooming behavior from honeybees, where they're trying to maybe potentially try and brush off those mites off their bodies. But the interesting thing is still, why is there such a greater chance to be finding varroa mite on the left side of the body versus the right side? And we still don't quite know why that is, but the theory behind it, or what people are hypothesizing, is that potentially it might have to do with the difference in the biochemical makeup of that fat body. So potentially what that means is, in different regions of the fat body, there might be slightly different makeup of that tissue that might be more or less attractive to the mite. So all of that is really quite interesting to see how mites are actually quite clever in terms of where they would attach themselves and where they would be feeding on honeybees. So there's definitely a reason why they are so difficult to see. But bringing that back to the function of the fat body, so we can now see, yes, mites feed on fat bodies, they attach themselves in certain locations, but what does that actually mean in terms of the effect on honeybees and their health? Because at the end of the day, as I think, and I hope I have already convinced everyone, that the fat bodies play such a crucial role for honeybees in so many different ways and functions, that they would have a big impact. Varroa feeding on the fat bodies, essentially feeding on the liver of the honeybees, must have a big impact on honeybees. And just quickly, what are the nutritional requirements and what does that mean? Because honeybees, to be able to have those tissue stores of the fat bodies and be able to function properly and be healthy, they need to have access to the best nutrition they can and all the nutrients they require. So honeybees get their carbohydrates from nectar, so essentially the sugars, and they require pollen as they need for any of the other nutrients. So whether we're talking about protein and amino acids, and very importantly, the protein content, well, in fact, that is true for all of the other minerals, whether it is proteins, fats, vitamins, minerals, they can vary depending on what kind of pollen we are talking about. So for example, the protein content of pollen can vary between 7%, often in wind-pollinated species, for example, protein content can be very low, up to plus 35, which is considered a very high protein content for honeybees. So the type of pollen really matters here, and the same is true for fats, fatty acids. So honeybees have essential requirements for amino acids and fatty acids, for example, that they have to obtain from their diet. And we know that some of our eucalypt species, for example, are low in isoleucine. So the only way that honeybees can compensate for those deficiencies is either by consuming a variety of pollen sources to make up for some pollen sources that might be low in some nutrients, or sometimes if a nutrient is not completely absent but it's still present in a diet, they might be able to consume a larger amount, but of course that has limits too. So the important thing to keep in mind is honeybees have essential nutrient requirements, but not all pollen types specifically provide bees with the same quality and quantity of pollen. But what does that mean again in terms of how does the nutrients that the bees need turn into the health of the bees and what they're capable in terms of performance? Because at the end of the day, it comes down to also what can the bees, how well as a colony can the bees perform. So as you can imagine, if the nutrition isn't right, if they do not have the essential like the fat body tissue nicely developed, and they're not able to carry out by any means the same function as if they were well-fed bees. So skinny bees really have a huge disadvantage, if you want to put it this way, in terms of like they're not capable. Honeybees that are not fed very well, that are stressed, are not capable to produce the same amount of honey. They cannot carry out the same pollination capacity. It affects their reproduction, specifically if nurse bees cannot support the nutrition of the next generation. Fertility in drones, we know it has a big effect on immunity, similar to us humans. If we don't have access to the nutrients we need, we're much more susceptible to disease, and of course it will also affect the longevity of honeybees. So there is a huge impact on individuals, but also on the whole colony. So now we are talking about a nutritional stressor. So honeybees can be stressed if they do not have access to the nutrition that they need. And this is where Varroa is celebrating, because that is where Varroa comes in. In fact, I should say the viruses are celebrating, because at this point in time, honeybees become much more susceptible to the viruses that the Varroa is transferring. So we know that, for example, adult bees that have been parasitized during development, so when they were still forming maturing in the cells, they show suppressed immunity as adults. They are also more susceptible to various pathogens. They have reduced longevity and reduced body weights, and they are not able to compensate for those impacts that they have been influenced by in that early development stage through, for example, pollen feeding or increased better nutrition later on. So it's kind of you're affected during development, and it is a long-term impact on honeybees that they experience from Varroa. And in fact, there is a study that has also looked at overwintering survival. So they have looked at they had essentially two groups of colonies, and one of those groups has been supported with additional pollen that was fed to make sure that they have access to all the nutrients that they need, and other colonies weren't fed, and that was in the autumn, so they just before the colony was going through winter, and they found that there was a significant impact of bees. Of the overwintering survival of colonies. So hives that were fed did perform much better compared to hives that weren't fed. But the interesting thing is that they state that it is, and this I think goes to show that it is again very similar to human environments in ways, because they found that not necessarily is there a difference between virus levels, like viruses are still around, but the bees had a much greater tolerance or resilience to fighting those viruses when they had increased immunity and good nutrition. And similarly to us humans, if we are exposed to bacteria, viruses every day, but it doesn't mean that we get sick all the time. So once honeybee colonies are stressed, whether that is through nutrition, which plays a crucial part, or for other reasons, they are much more susceptible to things such as viruses. So pollen feeding can also increase the longevity of workers that have been parasitized during development. So this was shown by another study. So there is a benefit to pollen feeding. In this case, the two studies have shown through prior to going into winter, but also through, okay, maybe we can cut those last two sentences out. So just quickly, I want to mention a study that I'm currently leading. So we're looking at supplementary feeding practices in honeybees, and specifically we are interested in the costs and benefits of supplementary feeding. So how can beekeepers use supplements, whether that is for a nectar or a pollen shortage, to support honeybee health, or to use it as a tool to grow hives or to increase hives productivity in preparation for pollination services, which are on increasing demand, or also for honey production. And of course, with Varroa being around, nutrition will become more important than ever. And this is a 4-year study funded between DPI and AgriFutures, and we are only just under 2 years into that, just over 2 years into that study. So that is still ongoing. So in summary, we can say that there is mites. Varroa mites do have a preference for where they attach to honeybees, or where they feed on honeybees, and specifically they're interested on the underside of the abdomen, because that's where the higher concentration of fat bodies are found. Fat bodies play a crucial role in honeybee health, and well-fed bees are much better equipped at dealing with Varroa and its associated viruses. So understanding and supporting bees in their nutritional needs is really key in having healthy colonies. And that starts by understanding the nutritional availability, so floral resources on specific apiary sites, but also colony needs during different times of the year, different seasons, and so on. So with that, I would like to say thanks for listening, and if you have any questions, please feel free to contact me. Thanks.
Our next presenter is Emily Nordyke, New South Wales DPI Technical Officer for Plan B, and she'll be stepping us through best practice for Varroa mite monitoring.
Hi, My name is Emily Nordyke. I work for New South Wales Department of Primary Industries in the role of Technical Officer Plan B, and today I'm going to talk to you about monitoring for Varroa. So you may already know about monitoring for Varroa, but we're going to go through some of the tips, tricks, and pitfalls today, so hopefully everyone can take something away from this talk. So you might ask yourself, why should I monitor? Well, monitoring is the basis for an effective Varroa control strategy. If you don't monitor, you won't know when the mites are at damaging levels in your hives. So it's really important to monitor consistently and effectively. And not to mention, it's also legally required every 16 weeks in New South Wales. But I want to start by giving you a note on visual inspection. So visual inspection, unfortunately, is not an effective method for Varroa monitoring, and this is because Varroa like to hide underneath the bees' bellies. As you can see from this picture just above me, there are two mites that have lodged themselves underneath the plates of the bees' abdomens, under abdomen. And so they really snug in there, they hide. So when you're going through your hive and doing your regular inspection, it's really hard to detect mites just on adult bees. So unfortunately, this makes visual inspection not effective. And if you do start seeing Varroa on top of your adult bees, then that means the damage has probably already been done. If you start seeing them on their backs, on their thorax, kind of on top of their abdomens, that usually means that the Varroa levels in your hives are already way too high and past damaging levels. So it's really, really important to use these other monitoring techniques that we're going to talk about today. So there are 5 main different monitoring methods that you can use. There's the alcohol wash, the soapy water wash, sugar shake, drone and capping method, and the sticky board method. And each of these methods have advantages and disadvantages, and also different specific situations where they're more effective or less effective. So we'll kind of talk about those today. But I do want to mention that I'm going to focus the presentation on the ins and outs of alcohol washing. Really, once you understand all of the steps in alcohol washing, then the soapy water wash and the sugar shake methods become much easier to go and learn on your own. So hopefully once we talk through the alcohol wash, you'll feel confident enough to tackle those other ones on your own as well. But I do want to note, we're going to kind of gloss over drone and capping and sticky board method today, just because those are used in slightly different situations. So they are good for surveillance or kind of just getting an idea of presence or absence of mites in your colony, but they aren't as effective for determining the exact percentage of infestation or your mite load in the hive. And that's what you need to use to decide whether to control for mites or leave your hives be for now. So really we want to zero in on the alcohol wash, the soapy water wash, and the sugar shake today. So let's get into the grit of alcohol washing. So it's the most accurate method for determining infestation levels where Varroa is already established. So in places like the US, where I'm from, Varroa's been there for 35 years. And so a lot of us use alcohol washing to get an idea of the infestation rates in our hives and make an action. And then the alcohol works to dislodge mites from adult bees. So that's how you're able to get the mites off the bees and count them in your wash, at the end of your wash. So what will you need for an alcohol wash? Well, first and foremost, you will need some type of wash jar. And this wash jar pictured here is something that you can pick up from pretty much any beekeeping supply store. So it's a really great tool for monitoring, and it makes it really easy and straightforward. You can also use different types of wash jars. You can use a simple mason jar and a sieve, but we're not going to talk about that as much today. We're going to talk about this particular wash jar just for simplicity's sake. You're also going to need a container for shaking bees into. So you can get a bit creative. Choice will contain the bees that you're shaking off the frame. So a baking pan, such as above, could work. A bucket. You could use a piece of cardboard even. Potentially, if you've forgotten something to use, then you can also use a hive lid, get resourceful. Basically anything that's going to allow you to shake the bees off and scoop them. You'll also need a 1/2 cup measure so that you can scoop the bees. And you can use methylated spirits, isopropyl alcohol, or ethanol for an alcohol wash. One other thing that you might find helpful is having a magnifying glass, using your phone to take a picture and zoom in on the results of your wash, or maybe your glasses so that you can see nice and close on the results of your wash. Those Varroa are really small, so this is something that you might find helpful. And in future slides, I'll show you kind of an idea of scale for Varroa in the bottom of a wash, so keep that in mind as we go forward. And finally, you'll want a bag or bucket for bee disposal. So let's dive into the steps of an alcohol wash. First, you'll place about 150 milliliters of methylated spirits or alcohol into your wash jar. Got a wash jar, for example, right here. So it goes just up there, but approximately 150 milliliters is what we're looking for. Then you'll open your hive and start going through the brood nest, and you want to find a particular kind of frame. So the ideal frame for a mite wash or any kind of mite monitoring, including the shake method, is to use an uncapped and capped mixture of brood on a frame. So above me is a good example. Obviously, we've shaken the bees off of this frame as the example, but you'll want that also pretty well covered with bees. And of course, remember to look for the queen. You don't want to shake her off the frame and scoop her up. So if you do find the queen on that frame, make sure to move her somewhere else or choose another frame. Next, you'll grab what you decided to shake your bees into or onto. In the picture next to me here, we've taken a metal sheet in half, and so it acts as a type of. I've done the exact same thing with a piece of cardboard and forgotten my other container to shake. So this is a really easy way to shake bees. Take that frame, and you're going to shake the bees forcefully right into your container. And once you've done that, you'll quickly scoop a 1/2 cup measure of bees. And the 1/2 cup measure is important because this is approximately 300 bees that fit into a 1/2 scoop. And then you'll take that 1/2 scoop of bees and put them into the jar containing the metho or the alcohol and screw on the lid. Now, these steps go kind of quickly. Once you shake the bees, you want to be ready with the scoop. Scoop those bees right out of your container and then dump them into your wash jar. And one thing I'd like to note is that with this jar, you've got a white insert that's already acts like a sieve, and your bees for the alcohol wash are going to go right into this sieve. Your metho will be in part of the container. So now that you have your sample of bees, it's time to do the actual shaking. So here you want to shake the bees vigorously for 15 seconds. And I like to shake the container in different directions just to make sure that the bees are well covered as you are doing the shake. Okay, so you've done that first step for 15 seconds, and then you can separate the bees from the container, from the liquid in your container, and inspect for the presence of mites. So you can either do that by taking off the lid and removing the bees with the white insert, or you can look underneath the jar and count the number of mites in the bottom. So that is a really easy way to do it. But if you do have a lot of mites in the bottom, it is also a good idea to separate those mites out between shakes because we're going to be shaking a few times. So you might also want to have maybe some piece of cloth to help strain those mites through in between washes. Now that's most important if you really have a high mite load. But then you're going to repeat step 7, shake again for 15 seconds. Check for the presence of mites, and repeat again. So you're shaking for a total of 45 seconds here. And if the mites are present, at the end of your wash, count the total number of mites and put that into your apiary records. Now, here are the pictures that will hopefully give you an idea of the scale of the mites. So this picture closest to me here shows the little red-brown specks of mites in the bottom of this special wash jar. And those mites are pretty small. They're about the size of sesame seeds, to give you an idea. So pretty tiny. And then the picture further over here shows the little flecks of mites in the bottom of a bucket. So hopefully that gives you an idea of scale as well. Another thing to consider is that the mites might be harder to see if the alcohol or the metho has gotten really dirty. So again, you might have to strain the mixture, or it might be best to switch out your alcohol between washes so that water doesn't stay really dirty and it makes it harder to see the mites. And there are also some things that look like mites, but they aren't actually mites. So really for me, the biggest mite trick, I guess, thing that looks like a mite that isn't, is usually a little red-brown flake of wax that makes it into the sample. So really when you're looking for a varroa mite, they're going to be very smooth, very ovoid, round, and not going to have like a jagged, irregular edge like a little flake of wax might. So you're looking for that perfectly ovoid little red-brown fleck. So then you'll want to repeat the required number of hive washes as per the requirements for your apiary, depending on the number of colonies that you have. And then at the end, dispose of dead bees by double bagging them and throwing them away. So now that we've talked about the alcohol wash, all the ins and outs, I want to give you a few notes on the soapy water wash. There are a few differences from the alcohol wash steps, but it's really mainly the same. We won't talk about those differences in detail today. That's something you can do on your own, just for the sake of time. But just keep in mind that there are a few differences. The main one being that you're using soap or a detergent. But definitely keep in mind that for a soapy water wash, you want to use a low suds detergent. If you have a really sudsy detergent, as you're doing that shake, it's going to get really bubbly and it'll be way harder to see the mites in the bottom of your wash. So here are a few notes on the sugar shake as well. There are really more differences in the steps from the alcohol wash in terms of timing, so the amount of time that you shake. There's a waiting period as well. So definitely something to dig into on your own if you're interested in doing sugar shakes. One thing to note, I think a major thing to note, is that it is not as accurate as an alcohol wash because that icing sugar is a little less effective at dislodging mites from the bees. So that's a big thing to keep in mind, but it is still an effective method to determine your mite loads. So still very much something that you can keep in your mite monitoring arsenal. One thing I also do want to note is that icing sugar can clump in high humidity. And if the icing sugar is really clumped, it's not going to be as effective at dislodging mites from the bees. And one advantage is that bees can be returned to the hive after you're done. So it's a non-lethal sampling method, which is really nice. So unfortunately, it's not as accurate, and an alcohol wash is really a great way to go. But here with the sugar shake, you can return the bees to the hive at the end. So we've gone over those main monitoring methods. Let's dive into determining mite loads. So really the whole goal of mite monitoring is to determine the number of mites per 100 adult bees. This is expressed as a percentage of infestation. And all of these methods that we've talked about, these three main methods I should say, the alcohol or soapy water wash and the sugar shake, all work to determine mite load in the steps that I'm going to talk you through in the next few slides. So once you've done your wash, count the number of mites in the sample, take that number and divide by the total number of bees in the sample, which should be about 300 if you've taken that hearty half scoop of bees, and then multiply that by 100 to get your percentage. But let's talk through an example of this. So let's say you find 6 mites after doing a wash on 300 bees. So take 6 mites, divide that by 300 bees, that gives you 0.02. Multiply 0.02 by 100, and then you get a 2% infestation rate, or your mite load. This can also be expressed as 2 mites per 100 adult bees. So I know it can be kind of a pain to have to mess with calculations when you're out in the apiary. So you can also use a good chart, a nice conversion chart like so. So you can look at the number of mites that you've gotten per 300 bees in your wash. So let's say 3% in this chart. And then right next to it is the percent infestation, your 1% infestation that correlates, or not correlates, but goes along with your 3 mites that you got in your wash, and on up, or in between as well. So we have, for simplicity's sake, used round numbers in this chart, but you might get 4 mites in your wash of 300 bees, and that would be a 1-point-something% infestation. So it's not always going to be clear cut, but that's why something like this chart can be really handy if you have it, you know, in your glove box, in your ute, or something like that. So let's just wrap up with some key takeaways. Monitoring is really fundamental in learning how to keep your bees healthy in the time of varroa. I cannot stress this enough. The beekeepers who consistently monitor throughout the year and take action based on their mite levels are definitely the beekeepers that fare the best when varroa is a part of their lives. And also, the alcohol wash, soapy water wash, and sugar shake are all effective monitoring methods. And at the end of doing your washing, make sure to determine those mite loads so you can take proper action if necessary. So thank you so much for listening today, and enjoy the rest of Field Day.
Pleased to introduce Elizabeth Frost, Technical Specialist, Honeybees.
Hi, my name's Elizabeth Frost, and I work for the Department of Primary Industries as the Honeybee Technical Specialist. We're going to talk today about managing your hives with Varroa. Varroa destructor is one of the most damaging pests of honeybees globally, and Australia now has it endemic, starting in New South Wales. It's restricted to currently a small area, the management zone as we know it today, and we're going to be discussing tools that any beekeeper in Australia can learn now and start practicing and implementing or planning ahead for. So what you see above me is Varroa destructor. These are 3 adult female mites collected from an alcohol wash. So we'll be talking about what it means when you find Varroa in your monitoring methods, either alcohol wash, soapy water wash, or sugar shake, and depending on the number of Varroa you find, what action, if any, you have to take next. If you take nothing else from this presentation, I'd like you to get your phone out and just snap this QR code, which will lead you to our new prime fact on managing Varroa in New South Wales. Managing a pest in a beehive is complicated. Most of us have experience managing small hive beetle in our hive, but Varroa is another kettle of fish entirely. So I'd like you to take a snap of this QR code, which leads you to a very thorough guide on the DPI website to manage Varroa in New South Wales. I'll be talking through various components of that fact sheet within this within this presentation, so we'll just get stuck into it. The thing on everybody's minds is, when I find Varroa, what do I do now? It's not a case of eradication anymore, we're transitioning to management. So the key thing is, as Emily Nordyke has said in her presentation, first off, monitoring for Varroa and understanding your levels. So what we've been seeing through the response is pretty large images of Varroa, but in reality, it's very small. You need glasses if you wear them normally to see small things. And what you might see as a result of an alcohol wash, say, is these 4 mites. So these are 4 Varroa mites, and you can see on this finger there's a couple of wax scales. This is a honeybee wax scale, which has been freshly extruded from a worker bee's wax building glands in her abdomen, and you can tell that those mites are very small. So you need to really make sure you have all your vision assistance if you use those tools. So what we've got here is 4 Varroa mites. What does that mean? This hive was monitored in the end of spring, so depending on the location, we could say this hive is in a stage of population increase for honeybees. Honeybees and brood quantity is increasing, and in line with that, so are mites. These guidelines we've got behind me are based off of the Honeybee Health Coalition group's Varroa control guidelines. So this is a group of researchers, extension specialists across North America, that's Canada and the US, that have derived these treatment recommendations. I will state a caveat. We're in Australia, not North America, of course. We're not New Zealand. We've got very different climate, nutrition, different pests, different viruses, luckily absent for deforming virus. So these recommendations are the current guidelines, and over time we'll learn about Varroa and how it performs in the Australian environment. So this is the guideline at the moment that you should follow. But as we learn more about Varroa's behavior in Australia specifically, it could change. So a good rule of thumb is, if you don't see any mites, you do not need to treat. What we're coming to now is a learning experience about chemical use in honeybee hives in Australia. And Varroa is a quick reproducer. In other countries, in the US for example, it has developed resistance to certain miticide chemicals because they've been overused in the beekeeping industry and in beehives. So our guideline is, if you're seeing 4 mites, you do not need to treat right now. If you see 0 to 5 mites in either spring or summer, treatment is not urgent. So I'll just harp on this slide a little bit. These are mite numbers that you might see from an alcohol wash or sugar shake or soapy water wash. That's collecting a sample of 300 bees, shaking them in whatever substance you're using, alcohol, soapy water, or sugar shake, and looking at how many mites fall out from that subsample from an entire beehive to guide you on assessing, are my mite levels at a point where I can wait a bit longer before deciding on some treatment, or I need to treat urgently. We'll come back to this slide again. So our key points of the year for the beekeeper living with Varroa is end of winter, early spring, is to get those mite levels to a manageable level. That will enable your hive to make honey in the season if the conditions allow it. Again, another important part of the season is the population decrease phase. So that's after they've made their honey, fingers crossed, conditions were good, and the honeybee population has peaked, also the mite population has peaked and is starting to go down. So you've got your 2 critical times of the year to get your mite levels lower, and that would be our population increase phase. So there's the least amount of brood possible coming out of winter. That's a good time of the year to either use a chemical control on your colony or insert some drone comb that you can attract mites to, trap them in there remove before the drone comb emerges. So you want to reduce your levels end of winter or early spring. If your mite levels become damaging through your population peak or the summer, you also need to treat then. And again, if your mite levels are at a treatment threshold during the population decrease phase, you need to treat again. So most likely in Australia, you'll have to treat a minimum every year 2 times for mites, either at the end of winter, early spring, and in leading into autumn to make sure you're protecting the winter bee population that's developing, and basically at any other point in the year when mites are at a damaging level. So we're trying to balance, I suppose, the mite population with these critical points in the beekeeper and the hive's season. So we want to protect the bees in spring that are going to be our honey production bees, and we also want to protect the bees towards the end of summer that are emerging and that will be our wintering bees. With Varroa management, we use these concepts that's part of what we know as integrated pest management. If you work with any growers, other farming sectors and livestock, they've heard about this style. So integrated pest management means that you're combining as many tools as you possibly can to suppress your pest levels below a damaging level to that hive's health or to its economic production. And we've got a lot of different tools in the integrated pest management toolkit. What is best recommended is to start with cultural tools. So these allow us to reduce mite reproduction, and those can be selecting for Varroa tolerance or Varroa resistance in our own stock. It can mean cultural beekeeping management practices such as splitting hives and requeening with cells, which creates a brood break. And if it creates a brood break, that creates a break in mite reproduction as well. Mechanical means that can be screen bottom boards. However, they're not a standalone treatment. Screen bottom boards are not enough on their own to reduce mite levels and prevent your hive from dying. So they can allow some mites to fall through the colony and not crawl back up, but it won't be enough without other treatments being employed. A good mechanical means of killing Varroa is by inserting a frame of drone comb in your hive. Mites are really attracted to drone brood because drones have a longer developmental cycle. The mites developing in the brood can feed for longer. So what beekeepers can do if their operation, you do your cost-benefit analysis. If it makes sense for you to try this in your operation, you'd pick that time of year when your colony normally raises drones. You'd put your frame of drone comb in, queen lays it up, the drone brood develops, and once it's capped, you remove that frame. So once you remove that capped frame of drone brood, you are removing this big stock of mites because they've been attracted to that drone brood. You've trapped them in there. Take it out, chuck it in the freezer, and dispose of it in a biosecure way. Just make sure you don't forget about the drone brood and it's starting to hatch out, because then you'll just create an even bigger problem for yourself. So once you've got to those levels that you've employed your cultural tools, your mechanical tools, and your hive will eventually get to that level where it requires chemical intervention, there are a number of tools commonly used by beekeepers, and those range from synthetic options to non-synthetic options. And they all have their pitfalls, but basically the beekeeping industry needs all of those tools to use as little as possible, but as much as necessary. With this IPM, integrated pest management approach, the ideal first is to use your non-chemical controls, but as soon as the mite population gets to a point that chemistry is needed, you look at your options and implement those according to the temperature in the field, are you making honey or not, how your hives are configured as well, and what safety precautions you need to take. So integrated pest management, it assists beekeepers to keep their hives alive, healthy, and productive. It assists consumers because it involves a safe use of chemistry in our hive to reduce or totally minimize chemical residues in our honey and our beeswax. And it contributes to benefits to agriculture, so we've got a ready and healthy supply of beehives to provide pollination for our growers. So talking about that top tier, common chemical control treatments for Varroa, here is a quick list, if you like. So we've got quite a few of those that are currently permitted for use through the Australian Pesticides and Veterinary Medicines Authority. That's our regulatory body in Australia for farm chemicals. So at this stage, we've got synthetic treatments permitted are, from the top down, Apivar, Apitraz, Apistan, and Baverol. And then our non-synthetic or organic treatments that are currently registered are Formic Pro and Apigard. So you can look up, just do a quick search online or on your phone for permit miticides or permit APVMA honeybees, and that will bring you the most current information on which chemicals are registered in Australia and legal to purchase and use generally by beekeepers in New South Wales. I will note one that's not currently registered, however we are urgently pursuing registration, is oxalic acid. So we're pursuing the product Apibioxol. Oxalic acid is a great treatment if your hive goes broodless, and spoiler alert, that's not many hives in Australia. So it doesn't travel through the cappings and kill varroa mite in the brood. So it is most effective when there's no brood in the colony. So it's really more of a late autumn or a winter treatment where you don't have brood in the colony at that time. So you're affecting all the mites that are lodged physically on an adult worker bee in the colony. Note of caution as well, oxalic acid, the two approved methods for using Apibioxol are vaporizing or sublimation and drizzling. So drizzling 50 mils in a full depth colony. And those two methods come with some pretty serious safety implications for the beekeeper as well as for bees. So oxalic is not recommended to treat a beehive over and over again. It's good use of chemicals and to prevent resistance by rotating different chemicals with different modes of action. So rotating your synthetics and your organics is a good idea. And not using any one treatment twice or more in a row. And oxalic is a really important one because it can have a negative effect on your honeybees themselves. They can build up oxalic crystals in the larval gut, and that impacts on larval development as well as worker longevity down the track. So make sure with any of these chemicals that are currently registered or hopefully going to be registered in future, you read the chemical label, which is the law, the safety data sheet connected with that chemistry, and of course the overarching permit allowing you to use it. And all those documents together will help you figure out how do I use this safely, how do I store it safely, how do I use it in my bees safely. With sourcing miticide, in New South Wales, the current procedure as of October the 16th is to report your mite results within the DPI Varroa website, specifically noting the number of mites that you've seen so that the assessment team can figure out, okay, they are at a level, they're seeing a level of mites, 6 mites per hive or greater, that requires treatment. DPI will contact you whilst DPI is the main supplier of miticides in New South Wales. However, in the near future, we'll have miticide being supplied commercially. So there's currently an approved list of miticide suppliers that you can search online. Just search the miticide you want to use, the letters APVMA, suppliers, and you'll find that current list. It's changing all the time, so I won't put it in this presentation, but if searching isn't your thing, just call up your suppliers that you usually use for beekeeping equipment and sources of nutrition and all that, and see if they're on that approved list. And if you're interested in becoming a supplier of miticides, you can get in touch with our farm chemicals specialist within DPI at this email address up here. So that's newsouthwaleschemical.coordinator@dpi.nsw.gov.au if you're a supplier of beekeeping equipment and you're not yet on that list of approved suppliers. So coming back to our treatment thresholds. Treatment threshold is a term for a level of a pest that requires treatment. And in the case of varroa mite, this is our steep learning curve that we're currently in New South Wales. So during the early stages of an incursion, we've got a large body of feral hives that are collapsing due to not getting actively managed to suppress varroa levels. So there'll be a period of time, it could be 1 to several years, in which feral hives will be crashing across the management zone at the moment in New South Wales, and also neglected managed hives or hives registered and looked after by beekeepers that just don't know what they're looking for or aren't doing their monitoring often enough to know that their varroa levels are at that control immediately stage. So again, as of October 2023, some areas of New South Wales are still in population increase, but others are gearing towards that peak population that summer period. So the recommendation changes between these two periods. We've got treatment control required at 6 to 9 mites in that population increase phase. And once you get into that peak population, typically honey production period, that threshold raises a bit to 9+ mites. And it's important not to treat less than that during the summer and honey production, because the tools we've got available to us at the moment are predominantly synthetic, and we want to be very careful that we read the label and the guidelines around its use during honey production, because what we don't want to see in Australia is chemical residues in our honey or in our beeswax. Okay. Again, you're looking for these mites in the summer. If you see in an alcohol wash 0 to 6 mites, 0 to 5 mites, you don't have to treat yet, but again, you want to be monitoring your colonies very often. Check out Emily Nordike's presentation on monitoring varroa and the specifics and pros and cons of the options there. During this, what we call the acute phase of the varroa infestation and establishment in New South Wales, we're going to be getting these random influxes of high varroa numbers potentially in our apiaries. So it really pays to be monitoring at minimum 4 times a year. That's a bare minimum, because you might find that your levels can spike, not based solely on the mites you have established in your apiary, but where your hives might be robbing out a colony that's crashing from mites and bringing a huge influx of mite numbers. So monitor, monitor, monitor. And if you're sick of alcohol wash and it's not that humid in your location, try out the sugar shake, because it's not lethal and it's very effective once you've got varroa established. So lucky last, I've got, if you didn't get the photo earlier of the QR code, this is the new page on DPI's website for managing your hives with varroa, and the QR code here will lead you right to the new fact sheet on varroa control in New South Wales that runs through all of the topics that I presented in this presentation around mite treatment thresholds, commonly used chemical control products, and the basics of integrated pest management for varroa for beekeepers. So please get reading, get looking at our videos. We've got an integrated pest management video on the website and fact sheets on safety data sheets and all that good stuff that'll help you learn about how to manage varroa in your honeybee hives.
Up next is Rod Burke, New South Wales Bee Biosecurity Officer, and he'll be talking about Varroa, viruses and biosecurity risks.
Hello, my name's Rod Burke, Beekeeper with New South Wales Department of Primary Industry. Today I'm going to be talking about Varroa, biosecurity risks from feral and unmanaged hives, mite surveillance and also virus introduction risks. 22nd of June 2022. We all know what happened. After their routine surveillance using miticides and sicky mats, the DPI sentinel hives in Newcastle were found to contain very small numbers of Varroa destructor mites. All hives at the 3 sentinel hive apiaries were euthanized that same night by Mark Page and myself. Some days later we still hadn't found any more mites, but went on a 10-kilometre drive away from the port and got dressed up. You can see what we found. We found some in the first 2 hives that we checked, then we stopped. It's quite shocking. These were high loads, 45 to 50 mites per hive from alcohol washing. So that was the start of the Varroa journey for all of us. We quickly learned that there were far more mites located 10 kilometres away from the port area than at the port itself. The DPI sentinel hives had discovered the expanding front, but the mites had not just originated from a port area incursion. So from extensive surveillance operations, we soon discovered that the mites were very widespread and that unfortunately had not been found previously by any of the beekeepers. What we also saw was just how poorly some of the beekeepers were managing their bees. It was clear that many were not following New South Wales beekeeping regulations and the code of practice. Many were not visiting their bees often enough and had too many hives to look after properly. Many could not manage weak or diseased colonies, so they spread disease. Many could not even clean up or bee-proof their dead hives or their other beekeeping equipment. So basically the level of poor, lazy beekeeping was unacceptably high. We saw a lot of sad stuff. We had hives that had been completely chewed out by bush rats and everything exposed, dead outs, fully exposed, and hives that only had 1 or 2 frames in them even. Not easy hives to manage. There were a lot of poor beekeeping and dead, exposed and robbed hives found. You look at this, not even any attempt made to stack everything up in a straight line, so of course the bees are going to get into that very quick. Some of it was basically just shameful beekeeping. If this is the best you can do, you really shouldn't be beekeeping. Sorry, but that's the absolute truth. You really can see why there were so many small hive beetle around. We had 3 great breeding years, very, very wet. Lots of beautiful breeding conditions and made even better by the fact that beekeepers were just giving it to the beetles, letting them breed up as much as they wanted to, causing huge, massive numbers of beetles to be coming out, going into everyone's beehives. It just makes it very hard when you're dealing with this. And now we've got this perfect union. We have this little mite which is slowly chipping away at hive health, and you've got these beetles that can come through and smash a colony in 1 or 2 days just when they get the right opportunity to do so. It's going to be very interesting to see how our beekeeping goes in some of the wet years that come. But so the situation, unfortunately eradication failed. We needed to try it. We didn't really want to live with Varroa mites, but unfortunately it's where we are now. So we're in a transition to management, meaning that beekeepers need to start learning how to manage their bees. We've got a management zone. We already know that these areas have Varroa mites in them, and so any bees that either come into those areas or are already in those areas will probably need Varroa mite management straight away. And then we've got a suppression zone where the mites are slowly going to be moving through those areas. So beekeepers in those zones need to start getting ready. Basically, at the end of the day, beekeepers have a new pest that they need to manage. So, as you can see from some of those previous photos, we already have a lot of beekeepers who already needed to improve their game before Varroa Destructor. So if they don't, then they won't survive after Varroa Destructor because it will take them out. They need to get much better at keeping their bees alive. So what is it about Varroa? Varroa actually kills a hive really slowly. It just impacts one bee at a time. One mite here, one mite there. It is not a problem for the beehive. But one mite will breed up and eventually become thousands and thousands of mites. So once established, it cannot be eradicated. You can manage the numbers in your hive, but you cannot completely eradicate them from the environment once it is fully established in an area. So beekeepers need to manage it in their own hives. Forget about what's around them, concentrate on managing them within your own hives. So you've got to do regular monitoring, and this means you need to kill bees. The best monitoring means that you're killing bees when you do that, and you need to get comfortable with that. You're better off to kill a small amount of the bees in your colony than to not do any monitoring at all and lose all of your bees. So there are different methods. Obviously alcohol and soapy water wash and sugar shake all test approximately 300 nurse bees if you do a half cup. Heaped half cup, about 300 bees. Nice, reasonably accurate number. It's a standard. And so these are the internationally recognised standard mite surveillance options that are used to work out what sort of infestation rate you have. How many mites per 100 bees. Then there's drone uncapping, and that tests how many drones. How many do you want to test from a hive? Basically that number is up to you. And then also, how does that result correlate with a wash result that's given you an accurate percentage infestation rate? Yes, drones can show you if you have mites or not, but you cannot find out exactly how many mites you have and whether it's time to treat or not just based off some drones having a few mites or not. It's just not accurate enough because the results vary considerably. One of these photos in New Zealand, they have a lot of mites. He pulled out a lot of drones. He found no mites on those drones, yet an alcohol wash was showing mites. Inconsistency all over. And you look at those drones there under the excluder, you've pulled them up, there are lots of exposed drones there, lots of different ages, but often you just cannot find mites in them. So it's very hard to get an accurate result. If we look at mites per half cup or 300 of bees, you've got 2 mites. Okay, you've got a couple of mites. Really, you don't need to worry at the moment. You have mites, yes, but you don't need to treat. You've got 3. Now you're up to 1% infestation. 3 mites over 300 bees is 1 mite per 100 bees, 1%. 4 mites, still it's not probably time to treat. You get to 9 mites, that's a 3% infestation rate. Most places around the world, you need to treat now. Maybe we don't need to yet without viruses, but if the viruses are about, you definitely need to treat at this point in time. So you need to do the regulatory monitoring. You need to be comfortable with killing some bees. And then you need to treat as required. When it's time to treat, spend the money, do the treatments. If you don't treat, this is what will happen 6 weeks later. And this is actually a test subject that we ran. We had 3% infestation rate. There are about 54 mites in this wash from 6 weeks later. About a 16% infestation rate. In a lot of places around the world, this colony would be almost dead by now. So that was by missing treatment by 4 to 6 weeks. So you need to treat because increased mite levels cause major health impacts upon the colony. And that brings out opportunistic pests that can see something that is good for them. Weakened bee colony, lots of resources, not very heavily defended. wow, this is going to be good. Robbing often occurs as well. So you need to learn to manage varroa, otherwise you'll be breeding up small hive beetles. They will take over colonies as soon as the critical mass of bees is low enough or they're stressed enough, the beetles will take them over. And you'll be spreading mites to others as well. Often as the colony gets weak and robbing occurs, all of your mites start to disappear into other colonies. They're called mite bombs for a reason. Mites blowing up everywhere. So you need to learn to manage varroa mites so you don't get mite drops like this on your sticky mat. There were thousands of mites on this sticky mat. This colony was very unhealthy. So there's more. As varroa slowly or quickly moves through the state, the feral and unmanaged bee populations will start breeding up mites. Feral colonies are often very widespread and they're really hard to find. You don't often know where they are, but there can be bees all over the place. Lots of feral bees and lots of little crevices, hiding, not always seen. And here are just a few pictures from some Newcastle Red Zone special operations that we did early this year. You can see we've got nothing and then there's a colony there. Colony in that tree, in that old house, that little black smudge up the top. That's a colony of bees, or it was, it's dead now. Down there in the internet pit, down there in that window, bees all through the wall cavity. Here we go, unlikely looking place, but guess what? Colony of bees living in there as well. And we've got this one as well, massive colony. And it had massive mite loading in it too. So feral colonies are all over the place. And we cannot manage feral colonies, so they cannot manage their own varroa mite numbers either. So we all know what's going to happen to that eventually. They're going to become massive mite bombs down the track. Their mite numbers will build up, and eventually a tipping point, hives dying off everywhere, and we've got the managed bees around, so those bees are going to be exposed to all of these mites. So basically this is a transition to survival. We need to keep our bees alive through this rough patch that is coming. You've got to work very hard. You've got a steep learning curve, but you've got to keep your bees alive. It's very, very important for yourself and for everyone else. Keep your bees alive. You'll lose some, but you'll be learning. You'll be learning to get better in managing mites. So the first thing is you've got to keep on monitoring. And I know the regulations say a minimum of every 16 weeks. Well, to be perfectly honest, during this phase, we should be doing a small percentage of our hives at least every 3 or 4 weeks. Say 10 to 25% of your apiary, do an alcohol wash, see what the mite numbers are doing. As soon as it hits a certain point, you've got to treat the whole apiary. It's going to be very expensive, but you want to keep those bees alive. You have to treat as soon as the threshold reading is reached, and that's obviously seasonally dependent. In spring, the bee population is building up. They'll build up very quickly, and so the mite percentage does not always get too dangerously high. At the other end of the season, it's the exact opposite. Very quickly, you can get very high infestation rates, and if you have that going into autumn, winter, there's a big danger that you'll lose your colony. They can get very unwell. So you've got to look at when you need to treat. Different times of the year, you need different thresholds for when you would treat. Don't be afraid to learn new things. This is a steep learning curve. There's all sorts of information out there from lots of different countries, lots of different personalities, lots of people that believe in one thing only. Give them a try. Follow what's legally acceptable. Use PPE if you need to, because some of these treatments are very dangerous. Just look at everything and see what is going to work for you. But at the same time, you need to manage small hive beetle, as they will take any opportunity to use your varroa weakened hive as a breeding opportunity. That's what they wait all winter for and spring for so that they can breed through summer. They want weak colonies so that they can breed up their numbers. Very importantly, follow the code. Manage your weak hives and bee-proof absolutely everything. And eventually it will get better. It's going to take a while, but the wave has to move through and then things will slow down, and then it's going to be managed colonies and very few feral colonies, and it'll be a lot easier to manage. So I'm going to talk about hives moving into the management zone, and I did that on the weekend. I've had bees 2.5 hours away sitting there, and I've just brought some back in. So the first thing you've got to realise is these clean hives will encounter mites very quickly. Okay, so you're moving from a safe zone where there probably aren't any mites into an area that's full of mites. It's going to happen, so mentally think about that. And don't freak out when you find mites. You know you're going to get them. So monitor them regularly. Treat when you need to. And enjoy keeping your bees alive. That's the thing about beekeeping. Keeping your bees alive. Know that you've got to manage mites to do that. So here's a little bit of small hive beetle and varroa monitoring and management. So mesh bottoms work best for a combined approach. So we've got 48 hours here since I put these in. I brought these bees back in. I gave them 2 days to settle down. I've got some hydrated lime in the bottom there, and I've probably got 50 or 100 small hive beetle that have been pushed through the mesh. At that point, I'm looking at something that I learned in New Zealand. They use core flute. They spray it with some cooking oil, and they use that like a sticky mat. So in this case, I've put small little sticky mats in. I've just put them in on top of the lime because I want to keep killing beetles, but I also would like to see whether any mites drop out. Then I put in miticide application. Obviously, this is a test that we're doing in surveillance hives. But you can see there, I've got 2 sets of gloves on. You've got to remember these are chemicals that you're playing with. You've got to minimise the risk to yourself. You've got to wear the appropriate PPE. Now, I actually prefer putting my strips through the queen excluder. A metal queen excluder will handle these strips very well. I don't really like putting them straight on the top bars because they can fall down and do stuff. So this is how I do it. And this was 24 hours later. You can see there's a lot of frass, there's some hive beetles, a bit of stuff has fallen on that sticky mat. And the results were that I found 2 mites on one of the 4 sticky mats within 24 hours. And here's one of them here. You can see there's a lot of rubbish and gunk on there. You need to wear your glasses. If you haven't got perfect vision, put the glasses on, get the magnifying glass, whatever you need to see that. And you can see a bit closer. That's our telltale sign of a varroa destructor mite. So within 24 hours of putting in miticide, I showed that this hive had already encountered mites in the environment. And it's only going to keep happening from there. We're now transitioning to management, and we all need to manage mites. And that means doing lots of alcohol washes. See what the results are. Hopefully you get small ones like we've got here on the left of the screen. We don't want those big ones. You know, that's causing colonies to become quite unhealthy. And you can see, very distinctive shape of varroa destructor. You don't want that, like I showed earlier, over 2,000 mites in that colony. That's not good beekeeping when they get to this stage. You need to manage it so you never see that. Because we're talking about viruses next. Varroa feed on the fat body, which is basically the bee liver. So this process weakens the bee and can introduce both bacteria and viruses into the bee. Remember, there's a hole. There's a hole has been put into the bee, and stuff goes in and stuff goes out into the varroa mite. So far, Australia has not detected the main deadly bee viruses found elsewhere, which are deformed wing virus and acute bee paralysis virus. If they arrive, which would be through the importation of bee genetic material or an incursion, then the number of mites required to impact bee health would drop sharply, meaning that we may need to be treating before we even get to that 3% infestation rate. At the moment we have a buffer, we can probably go a bit past that, but if we have viruses it's going to make things a lot worse. So it's very important at the moment. We must learn to manage our mite numbers. Thank you. Enjoy your beekeeping. And if you've got any further questions, please don't hesitate to get in touch.
Our next speaker is Mark Page, Training Coordinator Honeybees for New South Wales DPI, and he's going to talk to us about the Varroa response and the impacts on native beekeeping.
Mark Page, Honeybee Education Coordinator, New South Wales DPI and Tocal College, talking here on native bees. So there are approximately 1,700 species of native bees across Australia. The majority of these bees are solitary, and there are 11 species that are social or our stingless bees. One of our solitary bees is the ground nesting teddy bear bee, a beautiful bee, big and hairy, hence teddy bear. You just want to give it a cuddle. Another one is often seen in our gardens is the blue banded bee, again a ground nesting bee and a solitary bee. More of our solitary bees are your Nemea species, again ground nesting, and a lot of our ground nesting bees will have at times communal behaviour where their nesting site will be utilised by several female bees. And of course we have the cuckoo bees, this is a domino cuckoo bee here, that being a cuckoo uses the nest tunnels of other solitary bee species. Again, our carpenter bees, beautiful big bee, the great carpenter bee, hearing buzzing through the bush, nests in timber, so drilling a hole and tunnelling down. They will often use your grass tree stalks, your banksias, things like that, casuarinas, and being a carpenter bee, that's where it comes from, is because of their ability to tunnel using their mandibles. Others in the solitary bee species are our resin bees, our reed bees, and our leafcutter bees. So our leafcutter bees, often people will say, I saw a bee in my garden and it had a big green belly. So this is possibly a leafcutter bee, and it's using its mandibles, cut that half moon shape out of a leaf, and is carrying it back to where it's nesting. Wonderful creatures. So with solitary bees, there are some things we can do in habitat creation. We can do the blocks with drilled holes, we can do the lantana, we can use bamboo. Importantly that we create the right length, especially in your bamboos and drilling of holes and things like that. It is said that less than 70 millimetres long is not as popular, and you're looking at 5 to 8 mil holes with a length of around 120 plus millimetres is probably what is accepted by most of our solitary bee species, including our resin bees, our reed bees. So the other thing we look at, we could create, resources or pollen and nectar for our native bees in our garden. So plantings there's a lot of good material out there in publications that will give you indicators of which might be best. Not every plant that flowers has a pollen or a nectar resource that may be utilised by our native bees, but do some research there, lots of good information on what is good to plant. So our social bees, as I said earlier, there's around 11 species. The common species for us here with the New South Wales focus is your Tetragonula carbonaria and your Tetragonula ostroplebia. Now the location or the natural or geographical range, we generally say north of the Newcastle area, I say around Port Macquarie, is possibly good for ostroplebia, and south of Port Macquarie area or Newcastle area is probably more suited to your Tetragonula carbonaria. So their natural or geographical range, they kind of extend from up into Queensland, the temperate areas, which is really suits them. But once we start coming down below Port Macquarie, it generally tapers off, running down the coastal strip to around Bega. So locating these guys in a hive outside of their geographical range, there's a high likelihood that they may not survive. So be cautious of that one, do a little bit of research there. So the keeping of social bees. So a keeper of social bees, like an apiarist, coming from Apis mellifera, you're a meliponist from the Meliponidae. So one thing to think about in keeping social bees is the hive construction, especially depending on where your location is. So perhaps thicker wall structures and things like that might suit them more for the hot and cold. Location is important. We often look at things and go, well, I think that'll be the best place for these bees, but we need to do a little bit more research there because they are affected by really hot conditions and can be affected by very cold conditions. So our location is most important. Do a little bit of research if you're buying bees and locating them on your property. So keeping social bees, so conditions. So most importantly that we're aware of what's going on around us. What are the bees doing? Are they bringing in pollen? And similar to the European honeybee, you can watch your native bees at the entrance and you will see them coming back with extended abdomens, which usually means they're full of nectar. So available resource, these guys generally will only forage around 500 metres from their hive, so they're not able to do great treks similar to the European honeybee. So it is important to kind of be aware of your environment around you, what the bees are doing, especially in regard to drought that impacts. And I know in my area we are heading into drought, so I will not split my hive this year. There's some cautions you need to think about. Obviously fire can take out a lot of the resource for, in some cases, many years. And extreme cold or very cold conditions where the bees actually can't get out to fly to forage also can impact. So increasing hives. So hive splits need to be a little bit cautious. I keep my bees around the mid-north coast, and unlike the subtropical areas up into Queensland or north of kind of Port Macquarie, Mackay, we don't seem to have the conditions that are great for doing a yearly split. So we need to be very cautious on what we're doing. We can always open a hive and decide that it's not strong enough, and so we can put it back together. Doing a split without considering some of these things or the upcoming conditions, if we're going into drought, could possibly see you lose both sides of that split, which is not great for you and certainly not great for the bees. So think about our temperate and climate differences. So much, a great difference between, the Queensland, New South Wales border down to the Bega area. So we need to consider those if we're looking at splitting our hives and make sure we have a strong hive that we're going to split from. So pest and disease, like most things we keep, there are issues at times with pests. So one of the pests of native bees is the syrphid fly, really taping our joints after splits and keeping it so that that fly can't deposit an egg there between the joints, which will hatch and then crawl in and get involved in infest the hive. Another one is the forward fly, which again will impact on bees, especially after fresh splits or weakened colonies. Your Bambix wasp, which hovers at the front, is generally taking male bees, and I have actually witnessed them grab a bee, drop it, come back to then grab another bee. So the thought is there that it's picked a female, worked it out and dropped it and gone back to take a drone. So the way things evolve with each other is pretty smart stuff at times. And at times we are impacted by the small hive beetles, so doing a new split and protecting that entrance with a reduced entrance or a plug of propolis is often a good idea. One of the diseases discovered by Dr. Jenny Shanks is Shanks disease. It is a brood disease, a bacterial disease. It has been discovered, but I have not seen great impacts at this point in time. A common thing with native bees is what we call a fighting swarm. So it can be quite horrific when you look out and your bees are clouding and they're locking together and dropping to the ground with only one flying away and the other one deceased. This generally is a takeover or maybe a takeover, so the stronger colony will survive and either maintain that home or the colony that's come in to fight with them will take it over. Now we've had a few things about the response impacts on native bees. I've certainly inspected colonies through the Raymond Terrace and the Heatherbray area, and colonies that we keep here at Tocal and colonies at Patterson. At this point in time, these guys have not been affected. They're flying well, everything's going well for them, although we are heading into a bit of a dry period, so we'll see what happens there. As to our other native species, we have not seen any impacts, and possibly because, you know, for myself, I've been out a bit more and looking for European honeybees on resource, and I've seen certainly plenty of our native bee species foraging, so I think there's been very, very little impact on our native bee species, which is great. So pollination, this is another great thing about native bees. They are not the only pollinators. You know, there are many native pollinators, but native bees are the cutest. So in your backyard for your vegetables, your citrus and things like that, they are great. There is commercial advancement in pollination using your stingless or social bees, especially in your macadamias, your avocados and your berry production. And if you're encouraging native bees, you're encouraging beneficial insects in your garden, which will be other pollinators, moths, wasps, other bees, and flies, things like that. Honey harvesting. So there's a couple of methods in honey harvesting. The early methods were honey supers, where you punch them with nails and strain. Can get a little bit of pollen and a bit of resin contamination there where it tastes a little bit strong. But now there's some very interesting system with the printed cell system and the centrifugal extraction. Just a couple of cautions with purchasing. There have been people caught out where they're purchasing online. It's a scammer. There's interest in native bees and the scammer jumps in and says, yes, I've got some. You pay a deposit and you don't hear from them again. So be cautious there. Also, people selling fresh splits in areas that will not recover sufficiently to survive. So be cautious of that. Purchase off a reputable supplier. They're available. You know, check things out, talk to people, contact a native bee association, see their recommendations. You know, make sure it's suitable for location. You're possibly not going to take Tetragonial Ostroplebia down to Bega. Not a good idea. Make sure they're from a strong split, you know, and if you're doing one or the other. So if you're going to split it, if conditions are suitable, or if you're taking honey, just take the honey, don't split it at the same time. We can weaken our colonies enough that neither side will survive, so be cautious on that one. Conditions, as I say, we're coming into dry periods again. This will impact bees, so become a little bit more aware of what's around you. Of course, you can do garden plantings, and if you're in high residential areas, the bees will generally have access to other garden plantings, which is great. And beekeeping, whether it's European honeybees or native bees, is a learning curve. You know, we do learn from our mistakes. We will make mistakes, but learn from them, improve. Education is a big thing. It's something that we all should get involved in, especially when we're keeping of something. And the Native Bee Associations, become a member there, a local club or group. Obviously, combined learning, you can often hear the mistakes others have made and then not make them yourself. And that's where I'll finish. Introducing Dr. Cooper Shooten, Bees for Sustainable Livelihoods.
Right, hey everyone, my name is Cooper Shooten. I'm an academic researcher at Southern Cross Year, a research group called Bees for Sustainable Livelihoods, and we manage projects with the Australian Centre for International Agricultural Research in different Pacific Island countries where they have Varroa. And so today we're going to be talking about managing Varroa, what's happened in the Pacific and some of the lessons we've learned from other industries, its impacts on bees, beekeepers and pollination. Sounds like a whole PhD thesis or 10, but we'll try and keep it simple. Big thanks to Tocal for the invitation, New South Wales DPI, RBEEK, New South Wales AA, Amateur Beekeepers for the opportunity to be able to chat to you guys today. And it sounds like an awesome lineup, lots of great presentations and no doubt a lot of great information on varroa mites. So These projects, these are invested in by the Australian government, but they're also co-invested by partner governments overseas. It's not industry levied monies necessarily, but these projects have been going on with ACR in varroa mites. It's actually been investing in this for more than 2 decades. So some of ACR's initial investments are determining the difference between different types of varroa mites. There's 6 different species. 2 of them are reproducing on Apis mellifera at the moment, and building capacity for some of our labs in Australia, developing different methods, looking at the reliability and sensitivity of different detection methods, lots of different stuff. So just keeping in mind that varroa research has actually been happening in this country for quite some time. It's not necessarily something we're just starting now. We've got a lot of lessons we can learn from other countries as well. But in Fiji, a bit of a context here, varroa came in 2018. It was discovered the next year in both the major islands because there were people selling nukes across the countryside, and they weren't traced in lots of cases, and about a year after that they went to transition to management. So it happened pretty rapidly in Fiji. They determined they were unable to eradicate it. They did try to slow it down. They had emergency declarations, but yeah, it's pretty much now on every single island in Fiji. Interestingly, chatting to beekeepers there, watching this process go down, lots of beekeepers at the start said that they weren't really seeing much of an impact. There's some mites in here, but I'm still making honey. That's kind of what they were saying. However, after about a year or 2, the mite populations really started to get to some pretty critical levels, and that's when beekeepers were actually saying, I thought my colonies were looking good, and then they're coming back and they basically just crashed. And a lot of beekeepers started losing significant numbers of beehives, up to 30-40% in a lot of colonies. And at the start, the focus was really about, okay, what can I buy to put in my box? I want to kill these mites. What I'm going to go buy, I'm going to stick it in there. That was the primary sort of thought. And monitoring for those mites was an afterthought. It was only, more recently that people have been starting to tap into relating what percentage of mites am I seeing in this beehive to what's actually happening in the box. So, at what point do I really need to treat with different types of treatments, and when and how? So that was kind of an interesting process to observe, particularly at the start. It took a little while for suppliers to get access to and register and have supply of miticides, and there had been some issues that, in other countries, some miticides are not as valuable because they might have resistance there, and sometimes they'll just send them miticides that are just about to expire. So if you're ordering anything, make sure you check the expiry date before it comes over. So an interesting one as well was chatting to beekeepers, explaining that and also the Biosecurity Authority of Fiji, their equivalent, DPI, saying, look, we're seeing more AFB. Can you come out and check out what's going on? But really what was happening is they were observing a lot more parasitic mite syndrome. So that made looking for AFB pretty tiresome or confusing for a lot of beekeepers, because you're seeing this increase of AFB reporting, but not necessarily AFB confirmations or incidences, because the brood symptoms can look similar to other diseases. With this, obviously the larva isn't going to rope out, so that PMS is characterized by low bee population, abnormal brood patterns, sunken chew and cappings. The larva can be slumped in the bottom of the cell or on the side of the cell, but doesn't rope out. But I'm sure other people would be chatting about that during the day. There's still pretty limited rotation. Getting there with it does take time. At the start, we're seeing a lot of beekeepers going out to Bunnings, their equivalent. They don't have Bunnings, unfortunately. A lot of people just going to Bunnings and seeing what they can stick in beehives. So it can be pretty concerning when you see that. I know beekeepers, we want to, obviously, the cheaper any management solution, the better, but there's also some things that could be optimized for the better of people, bees, and killing those mites. So later on, people started talking after that about, they started learning about resistance issues and that sort of thing. So it's a bit of a continuum and a journey and that sort of thing, observing what was going on in Fiji. In Papua New Guinea, they don't have small hive beetle, they don't have American foul brood disease, but they do have 2 different mites, Tropolaelaps mites. There's 4 species of Tropi. 2 of them are reproducing on Apis mellifera, and managing these 2 mites in combination is not a lot of fun. We've trialled some experiments in Papua New Guinea around, how effective some of the treatments are that they're using, and different mechanisms for beekeepers to use non-chemical control methods, because a lot of people up there don't have access to money or those different types of equipment and the extension to be able to use them appropriately and that sort of thing too. But I'm not going to chew your ear off about that research. If you're interested in it, you can send me an email and I'll send you a copy if you're having any difficulties downloading it off the web. So lots of different pests and diseases of honeybees. You guys know about, more than 40 different pests and diseases. We need to be able to identify them, monitor them and manage them. It's really, really critical. But the focus now is Varroa. For a lot of people, but it's important to keep in mind that we're managing Varroa in the context of genetics and nutrition and ultimately in combination with many other interacting stresses, including other pests and diseases at the same time. It's important that we just sort of keep those things in mind. So as you guys all know, we are advocates for this. If you weren't, you wouldn't be sitting here listening to me right now. But, it's about those beekeepers around us too, that we're communicating. We are the advocates for this, that you can't just stick bees in a paddock and expect them to be healthy and happy and productive, particularly now that we have Varroa. I mean, it's not the end of the industry, but we will see the exiting of a lot of lazy beekeepers. That goes for pests and diseases that we do have in Australia and these ones here that we do not. So as I was mentioning, there's different types of mites, Tropolaelaps mites, and we've got Uvaroa sinai on Apis florea, Apis cerana, and actually that's in far north Queensland, but not in lots of other places. Mites, different types of viruses, that's a picture in the top right, and other types of mites. So we need to be aware of these. I think Sammy Giggins, legend, is going to be talking about some of these exotics as well as other endemics, but obviously we're going to be focusing on this one. So as you guys know and probably heard already, but these mites are basically feeding on the brood and the adult bees. They spread viruses, they make them weak and deformed, they don't live as long, the population gets smaller, they don't produce much honey, and if you just leave them there, they'll die. There's actually lots of viruses as well. I think Rod Burke's going to have a chat to you guys about viruses, but just keeping in mind there are some that we have here in Australia already, the ones with the asterisks there, and there are also ones that we do not have. They're the ones without the asterisks, and the ones that are red are particularly bad for bees, so we need to make sure we're keeping those out. How bad is a mozzie in comparison to how bad is a mosquito with malaria in it? You're talking about a pretty different problem. And interestingly, the viruses actually might influence what our economic thresholds are. So what percentage of mites infestation are you seeing that's actually causing that economic damage is going to be influenced by the amount of viruses and the prevalence of them and their distribution. So something to keep in mind for beekeepers. Killing those mites doesn't necessarily kill those bee viruses, so that's something to keep in mind. When we're talking about the effects of Varroa on honeybees, usually we just say that they're chewing on the fat body of the bee and they're spreading viruses, but they're actually doing heaps of other crazy things. There's some publications there that you can follow up. To summarize that slide, this is that slide summarized. So they're spreading, activating, exacerbating these viral infections, causing physiological deformities. They don't live as long. They're, more susceptible to pesticides in the environment because bees don't have a liver. They use their fat body tissue for that. They've got a diminished immune response. They're lighter. They're impaired. Their orientation abilities go down. Their ability to communicate and produce royal jelly goes down. So these mites are doing lots of different things. It's not just feeding on them and spreading viruses. It's quite difficult to be a bee with all these things going on. But in saying that, the effects of all of these different things and the effects of Varroa on individual bees do not necessarily translate to similar effects on the whole colony, because as we know, honeybees are a super organism. They're social creatures. They live in complex interactions with one another and the environment. What we do know for the research is that when you've got lots of mites, your colonies are going to be significantly lighter, less bees, less food resources, particularly coming out of colder winter climates, for those of you in colder areas. It can exacerbate and spread diseases between colonies, because if you've got really weak colonies, well, they're more likely to be robbed out, and if they've got AFB, it can spread around. We know that if you just stick them out there and leave them in a paddock, they will die between 2 to 3 years maximum. High infested colonies significantly increase their chances of dying. Well, that's pretty obvious, but in combination with other things, it's not ideal. Large proportion of the worker population are just sitting in there picking their noses. They're not feeling too good. They're not going out for work. you can have significantly higher rates of queen supersedure. So when you're requeening, you can actually have quite a lot of difficulties, significantly higher rates of that not being successful. So having low mite numbers before you actually requeen is going to increase your chances of that being successful potentially. And as we chatted about parasitic mite syndrome, main thing there is it can, for some people, make AFB diagnosis a little bit more difficult. Some of the effects on beekeeping businesses. So there's been a fair few studies. Most of these studies are pretty old. They look at lots of different types of information, so we take them with a bit of a grain of salt. But generally, we know that Varroa is going to increase significantly the amount of management, monitoring, and replacing costs associated with Varroa losses. So having more people out there knowing what's going on in those boxes in terms of where your mite numbers are at. In New Zealand, there was a study there, 2008. They had increased costs of about $40 to $50 per hive. So translate that to today's dollar in AU dollars, that's about $50 to $60 a hive. There was a study that showed for the average size Australian bee business, costs could increase by 30%. Keep in mind the average is probably perhaps not necessarily the best number to use. I mean, the average number for bee businesses in Australia is 300, so or 299 in 2018. So if you've got that many beehives, that number kind of makes sense. But for other players, obviously take that number with a grain of salt. In New Zealand and the US, we had a 50 to 60% reduction in hobbyist and part-time commercial operators. And that's pretty important because they play a big part in the industry, having a lot of fun doing what they're doing. It's an important part for some people, part of their income, and it's worth about $173 million to the economy. However, impacts on honey production are actually pretty difficult to discern. There's not a lot of research out there that shows that a clear pathway when these mites turn up, that you've either got major declines in honey production. Some actually show that there's actually little difference, which is quite interesting. So what we do know is that Varroa is a leading cause of colony losses globally, particularly in USA and New Zealand. We know that management options can't eliminate Varroa, so once we got it, we just got to keep it at levels that are below those damaging economic threshold levels, basically. So once it's there, these treatments are, if you read the packet, it doesn't say 100% effective, it says something like 98% effective. So you've still got a few mites in there, and obviously a lot of them move around through drift and beekeepers moving stuff around. So in New Zealand, they've seen hive numbers remaining pretty steady when after Varroa mites came, but the number of registered businesses was cut in half. So that's pretty interesting. In the USA, there was little change. So this is actually a bit confusing because if you're reflecting on what I'm saying that Varroa is the leading cause of colony losses globally, but there's not clear research that says that beehive numbers and honey production change that much. Why is that? If Varroa losses are so high, why doesn't this have a dramatic impact on hive numbers and honey production statistics? Well, believe it or not, beekeepers are really good at making bees. They make splits, they make nukes, they're making queens and packages. And the reality is that most of beehives and honey production, most of beehives is owned by a very small percentage of, highly skilled operators that are going to be out there learning a lot about these mites and applying different integrated pest management solutions. But in saying that, you still got to replace those colonies that are lost. And that means a significant increase in your costs of production. So some of the impacts on pollination industries, you guys know, we've got 35 pollination dependent crops in Australia in particular, so 65% of our crops, we need some honeybees there for those growers to remain viable and have optimised production systems. And that's worth about $14.2 billion to the Australian economy. Keeping in mind that study was done 5 years ago, and that study was looking at data from 2015. So that number is actually probably much greater now. There's been a lot of crops going in the ground since then. There's actually very few scientific studies investigating the impacts of Varroa on pollination. It's pretty hard to do studies that look at mites and then how that impacts on bees and then how that impacts a colony and then how that impacts on a crop. Actually, quite a long bow to sort of draw there in terms of connections. It's pretty hard to research that, but the research that is out there, and there's probably a lot more than what I've cited here obviously, but generally speaking, bees from colonies that are highly infested with mites are going to be less efficient pollinators. As we know, they're not living as long, then they've got impaired orientation abilities, possibly due to shorter flower visits and lower tendency to collect pollen. So there is a few studies out there that show that. You've probably heard already, the varroa is going to progressively kill about 95% of Australia's feral bees within the first 3 to 4 years. We've seen that in other countries as well, particularly in the USA and in Europe. And as a result of that, lots of growers are out there getting a free pollination service. It's incidental pollination, and as a result of that, there's actually perhaps a low level of awareness around the importance of pollination by honeybees than might be expected, given it's important because we've got so many feral bees in our Australian environment. We've got beautiful flora and great environment for bees to support themselves, essentially. So when those feral colonies die, growers are going to need more bees than they already have to supplement those losses, and some marginal growers that are not seeking bees at the moment will probably need to start getting bees in. So what does that mean? Greater demand, lower supply, increase in cost of production of pollination services. A couple of studies out there show that the price paid for commercial hives will raise between $70 to $110, so that was in 2010. So there's your 2023 stats, anywhere up to $160 increase. There was another study by Ryan. I actually emailed the guy the other day to get the report. He said it could go up by a total of 440 per hive. So obviously very variable depending on crops, but obviously the key thing here, and as seen in New Zealand, is that it's going to increase, particularly in the first 5 years, anywhere up to 100% in those costs for rental hives and pollination. So what we know, prices paid for pollination is going to go up, no doubt about it. There's a lot of different crops. Different crops have different dependencies on having honeybees there, and obviously those crops that are more dependent are going to need more bees to be on them, essentially. And then this slide just looks at the value, so the value of pollination dependent production, so some of those top crops, and obviously almonds, apples, oranges. Sorry guys that the text is so small here, but yeah, just keeping in mind when you're thinking about your business model, which crops are you visiting, which crops are most needing honeybees there, and which ones, obviously there's a lot of growers that are out there, essentially. So both beekeepers and growers are going to need to be proactive. So I just wanted to emphasize that, you know, we've seen in New Zealand, for example, that some of the stone fruit growers there really didn't pay to have more bees on their crops. And as a result, they waited until their business models were almost non-viable because they just weren't producing the crop that they needed to get those margins that they required. So both beekeepers and growers need to be proactive in ensuring they've got adequate pollination services happening. Just to switch the tone a little bit to a topic called chemical resistance. So I just wanted to show you this. This is after Jack and Ellis paper that came out in 2021. Just really highlighting the importance of being aware, basically, that chemical resistance is a real problem. So on the left-hand side, you've got a number of different countries there, and at the top, you've got different types of key miticides that are used to kill mites. And you can see that in some of those countries, you've got people using those chemicals and paying good money for them, putting them in a beehive, and the varroa mites just basically laugh at you. They're not going to be as impacted by those treatments as they should be. So that's obviously not an ideal situation. If you don't believe me, these are some of the publications that you can follow up on, and that's just where data exists. So this is potentially happening in other countries and at a greater extent, but it's just not necessarily recorded. What causes chemical resistance? Using unregistered chemical controls, or stripping out to Bunnings and getting whatever you think you want, just putting in a beehive. Using less than the recommended dose, and I'll just cut this strip up and divide it between different hives, save some money. Using the same chemical every time, so just using that. And keep in mind, the chemical class, not the name of the packet. The packet or the brand could be different brand names, but they're using the same chemical. So make sure it's the chemical that you're actually, the active chemical in there that's actually been changed. And not removing strips, so just putting them out there, basically just not reading the instructions on them. It is pretty simple to just follow the instructions. I do anticipate beekeepers are going to trial and experiment things, particularly for some of the organics, which is great. But yeah, there's a lot of safety considerations for both people, bees and bee products that really needed to be taken seriously. Otherwise you can impact on those things. And essentially the process is that, if you're under treating or doing any of these things, you'll have some percentage of the mites that just get a little bit of a taste for that chemical, and they don't die, and they go on to reproduce, and you just essentially end up with more mites that don't care about those chemicals. So something that we hopefully can avoid here in Australia. So I put it to you all, I asked you guys the question, what will the known varroa chemical resistance look like in the year 2033? Because in New Zealand, there's resistance been shown for all 4 of those major chemical classes. These other countries in the Pacific, including Australia, we don't know yet, but it's up to us as beekeepers. So essentially, resistance issues are real, and it's up to us. Key take-home messages: you can't expect honeybees to be healthy, happy, productive if you just stick them in a paddock. You guys are the advocates for this. I know you know that, but we need to communicate that to other beekeepers. There are major pests and diseases in and around Australia, and we need to be able to identify, monitor, and manage those pests and diseases. So just because we got varroa doesn't mean we take our finger off the pulse with our biosecurity and all the great work that's been done there, and also, importantly, us learning about these things as individuals. Killing bees doesn't kill bee viruses. Varroa will increase the cost of production, but honey and colony numbers may remain stable. They might. Varroa will increase the demand for bees, and therefore the prices paid for pollination, no doubt about it. Beekeepers and growers are both going to need to be proactive. And sorry about that varroa mite being on top of that text, but varroa is not the end of beekeeping, and it says there, unless you're lazy. It's just a new way of beekeeping. Anyway, lovely to be able to communicate and chat to you guys about varroa. It's something I'm very passionate about, and I hope you're all having a great day.
Up next is Anna Noon from the Groundswell Collective, and she's going to talk to us about creating urban wildlife oases with tiny forests.
Hi, and thanks for inviting me along today to about tiny forests and the role of supporting biodiversity and pollinators. I've been leading a tiny forest movement here in my hometown of Lake Macquarie in conjunction with 2 not-for-profit community organisations, the Groundswell Collective and Relief Lake Mac, both of whom are dedicated to building collaborative partnerships to support and increase local tree canopy, habitat and community resilience. I'd firstly like to acknowledge that I'm recording this from the traditional lands of the Awabakal people, and I'd like to pay my respect to elders past, present and emerging, and extend that respect and warm welcome to any Aboriginal and Torres Strait Islander community members listening in today. Before I launch into what tiny forests are, I just wanted to give you a little bit of background of how I got into this. So back in 2019, my family and I moved onto a small acreage on the outskirts of Lake Macquarie. I'd grown up on a sheep and cattle property in the New England when I was young and was always keen to get back on the land. And what you're looking at here is when we purchased our property in October of 2019, it looked like that photo on the left. It was lovely and lush and green. And by the time the property settled and we moved in, it looked a little bit more like that photo on the right, which was December of 2019. And from there, it continued to get much drier and much hotter. And we spent our first summer surrounded by bushfires, making emergency plans and running very low on water, which was a real baptism by fire for my children of life on the land. And then you will all recall, as vividly as I do, I'm sure, following the fires came COVID and this whole time of kind of rethinking for lots of people. Between the move and those few events, it really prompted a personal journey for my family and I about living more sustainably. We started with really small efforts, and I've included some pictures there of things like worm farming and composting and growing our own veggies. And I also joined a number of community groups to do more out in the community. Now, during that time, I was also doing some tertiary academic studies into sustainable living. And part of what my studies included was getting us to look at local solutions to really large wicked issues. So those complex and difficult to solve problems that exist in our local environment, but also globally. And it was at that time I really started to deep dive into biodiversity loss and learn a lot more about how human activity is leading to mass species extinction, how we're upsetting the balance of nature and making our ecosystems fragile and less resistant to disruptions. I learned a lot about how biodiversity and nature is critical to our survival, obviously providing the air we breathe, pollination for our crops, and giving us the food, fibre and fodders we rely on. And I learned a lot about how we urgently need to transform and make big changes to restore and protect biodiversity across both our local environment and more broadly globally. From a local perspective, here where I am in Lake Mac, we're like lots of coastal urban areas. We face the intertwined challenges of climate change and accelerated population growth. Our population of 205,000 is set to grow by another 25,000 people over the next 13 years, which will create an increased demand for housing. And obviously, as the number of people living and working in our city increases, so does the risk to urban nature. We really need to plan to take proactive measures around the lack of urban green spaces, which is resulting in fewer habitats for wildlife and the amplifications of climate change, such as flooding, pollution, and heat stress. So I was deeply thinking about these issues, doing a number of different things to try to address that both at home and out in the community. And I'm sure, as you will know and are all too familiar with, then more recently we had the outbreak of Varroa, which posed this increasing risk for pollinated services and biodiversity support locally. So what I did is I went off and I did some kind of deep dive into academic literature around international studies of projects that have undertaken evidence at base biodiversity support and pollinator support. I also went out and spoke to a number of our existing greening groups about what they'd like to see if we were to run some community projects that would look at increasing green space in urban areas and biodiversity support. And these were some of the things they came out and said. They wanted projects that inspired hope, that provided education to people who may not have a deep understanding of the importance of these projects, something that was more than just planting trees, but was leaving a legacy behind. And that's when I came across what's called a tiny forest. Just a video for you. Just hang on a sec, I'll just screen across because I think this video says it better than I can. So, what I learnt about tiny forests was that they were this densely packed patch of native bushland planted using organic soil remediation in an area about the size of a tennis court. And there are examples of these all around the world. There was lots in Europe, they had started in Japan, they had been expanded into India, they'd gone through Tehran, and a whole world of different global areas that had adopted this method given the evidence around them. So they were this international movement with over 3,500 tiny forests, both worldwide, and they were in both urban areas and in rural areas. And as that little video out, they were developed these botanist Eventies, who was a specialist in studying restoring natural vegetation on degraded land, and his name was Akira Miyawaki. And he looked at ancient forests surrounding Shinto temples in Japan and developed this theory and method to accelerate forest growth and mimic a mature forest. Now, this slide has quite a lot of details on it, but what you can see on the left is kind of a traditional way of a climax forest developing over time. So you take the climate, the topography, and the soil, and that influences what happens if humans impact along the way and bare land is there. Then it will take up to 200 years. You can see down this side, it'll take up to 200 years to reach this final ecosystem. And what Akira Miyawaki decided was he could expedite that process by adding organic material to the soil and densely planting forest trees from this climax variety that would appear there naturally at this point, and he could create the same result within about 20 years. Now, this slide shows you a similar process, just in a different way. So if you have a piece of degraded land, whether it's been burnt by traditional methods or bushfire, or if we clear it and we let it naturally regenerate, it will go through these stages. First, you'll have your pioneer plants, and then your grasses, your shrubs, our short-lived trees like our acacias that often come in first, grow quickly, die quickly. And then over time, you'll get to this point where you have a climax forest. So what Akira decided was he could improve the soil, plant these species, and they would grow rapidly, saving all those years and time in regeneration. And what they found from studying these tiny forests was that they were 100 times more biodiverse than the existing land that had been there before. So if it was grassland, they were 30 times denser than monoculture plantings, and they grew 10 times faster than traditional planting methods. So with that in mind, I set about developing a pilot project here in the Hunter, which was called the Lake Mac Tiny Forest. And it had this vision. It was a vision to be an innovative, community-led solution which aimed to support local biodiversity, protect and enhance our environment. It was based on that research from global examples about the benefits to both social and environmental benefits to the community, and that would be part of this worldwide movement that helps support biodiversity, trap stormwater, and assist with combating climate change. So it took about 2.5 years to get the approvals and the piece of land. So we had a piece of public land that council donated to us, but we then set about undertaking the steps required to build a tiny forest. So we did detailed soil studies, we loosened the earth and added organic materials like compost and manure. We fitted, you can see there, we fitted some timber trunks around the outside to provide some weed protection, and then installed an outdoor learning classroom there for people to come and visit the forest. And then we had 60 students from Teralba Public School come and help us plant some of the trees and 70 community members that came along, and in a small patch here of 340 square metres, so a little bit bigger than a tennis court that you can see here, we planted 750 canopy trees. We also have our second project under development now that's called Tiny Forest, and this is going to be planted at the end of October. And this project is a direct varroa mite outbreak. The land is a private block that belongs to a previous apiarist who had 10 hives for a varroa response, and they're now turning a portion of their property into an urban oasis for native pollinators. One of the really exciting things about Tiny Forests is, along with the program, we have volunteer tree keepers who offer up their skills and services to maintain the site for 2 years. And after 2 years, those tiny forests are self-suppressing, so the weeds suppress naturally as the canopy grows, so they don't require any human intervention after 2 years. And we undertake citizen science studies alongside of it. So we look at the benefits to thermal comfort. So obviously having a patch of trees helps to reduce urban temperatures. So we look at the thermal comfort differences inside the forest and outside the forest. We look at the positive impacts for flood mitigation, how water is stored through the soil given its improved health. We study the biodiversity, looking particularly at pollinators that are attracted to the site, as well as soil dwellers. And we do these studies every 6 months to see the improvements over time. We also measure carbon absorption, and we do some studies around social and health improvements from being part of the program. So tiny forests are more than just plants and animals. They're about providing a place for people to connect. They provide inspiration and hope about nature-based solutions to climate change, things people can do to support biodiversity, and these little patches of dense habitat have been proven to provide really significant support to biodiversity. We undertake education and information sharing. We often do tours and talks about tiny forests. We invite school groups and any groups along to come and learn about what we're doing and why we're doing it. We build partnerships and collaboration between the community and local businesses, and everybody kind of pitches in to help out. And they're a really great example of grassroots actions that can be taken to mitigate the impacts of climate change. Now, I think that's my time up. So if anyone is interested in learning more, you're welcome to jump on our website at thegroundswell.org, or you can email me at anna@thegroundswell.org.au. And if you have a little patch of land that you think might be suitable for a tiny forest, we would absolutely love to hear from you. Thanks for having me.
Our next speaker is Sam Jiggins. New South Wales DPI Bee by Security Officer Surveillance, and he will be talking to us about exotic and endemic pests and diseases of honeybees.
Good afternoon, everyone. My name is Bee by Surveillance, and I'll chat and quick background to cover some exotic pests and so far interesting, and hopefully I'm not with what I'm about to talk about. It's not too doom and gloomy. We'll kick it off anyway. I'll give you a little bit of a background of my my experience in bees so far. I started out beekeeping out in the central west of New South Wales from a very young age, around 10 or so, with my grandparents. So I'll start with a little bit of a tribute to those legends. Joan and Peter Stone Street out in the central west, New South Wales. They got me into bees from a very young age. There's an image there of my grandfather with a flow hive in his 90s that he was managing. And I would come in and check on it every now and then just to make sure everything's all right. But yeah, these two people got me into bees, and they gave me the bug, so to speak, that everyone talks about. A good friend of mine says beekeeping's not a profession, it's a disease. Well, I got the disease because of these guys. So thanks very much. But I love it, love bees, and I love everything that's involved around it. My interest and my thirst for knowledge around bees just grew as I grew older. I'm a diesel mechanic by trade, worked for Australian Defence Force as well in the Army. So I've always had a passion for agriculture, and bees have always been in the background for a very long time. So I decided I wanted to get a bit more serious about it after working for the local ABA club, being an apiary manager, and I wanted to get more experience. So I went and did the Cert 3 in beekeeping out of Tocal under these people, Doug Somerville, Danny Lloyd Pritchard, and there's a very young, fresh Elizabeth Frost straight off the boat from California. But we were the first cohort, my wife and I. There's my wife Bianca in the background. And interest for the environment grew, watching and monitoring all the eucalypt species and their flowering events. And it just kept escalating and escalating. The disease kept taking over and consuming my life. Doug was a big advocate and mentor for me, as was many others. The local New South Wales AA branch, Cole Wilson. I've been a member of the New South Wales AA for a good 8 years now, and 8 or 9 years. And they encouraged me to go and get some industry experience. So off I went and worked for Lockwoods for a while out in the Central West, the Goldfield apiaries. There's an image of me out there doing honey production. Also queens for a few queen breeders in the Newcastle region. Jamie Baggs. But there's some nuke boxes there that I was part of developing. Drove trucks down for almonds. Done some, that's me at almonds probably 7 years ago or so. So I had a quite a decent level of experience. This is a little collage of just how things just got a little bit out of hand, and I just got into it even more. Image in the centre there is my wife and I in our shed we were living in at the time. That's our living room. Building beehive boxes. With my military background, with a dog, we trained a dog to successfully scent detect AFB. In hindsight, I probably should have had it successfully detect Varroa, but this is just giving you an indication of how things just got. I've just been totally wrapped up by bees, and my passion and drive for bees is like nothing else. There's nothing else that gets me out of bed to go and learn, and to be employed to do bees full time is an absolute privilege. So there's the image in the top left there is myself doing some instrumental insemination under Casey Cooper and Elizabeth Frost. This is other things, just escalating once more into bees. My commitment. Above there is our extraction shed that we built very recently, and there's my face looking how chuffed I was. I was pretty excited about having a big shed, but we spent a fair bit of money. We've escalated to having a truck, and all the other things that are involved with bees. So life in beekeeping was pretty fun, or it still is, and quite peaceful and interesting for me, with very little problems apart from having an empty wallet from spending all the cash. Any spare penny I have, I spend it on bees, just because that's what we do, isn't it? But then, this happened all of a sudden, the 22nd of June, the detection of Varroa destruction. It just turned up on our front doorstep. That's about a week after it was publicly announced. Going and doing some volunteering work in the Newcastle region, where I first came face to face with Varroa destructor, and that was very confronting. And from there, it's I started doing volunteer work, and then went to paid work as a team leader within the Varroa mite emergency response. And that's what I've been doing for the last 12 months, been actively going out and looking and searching for Varroa. Found a lot of infected premises in the last 12 months. And then I applied for the role under the bee pest surveillance program, and that's where I ended up. so far. This is where I am as of a week ago. I was working for the National Bee Pest Surveillance Program. So the National Bee Pest Surveillance Program is funded by Hort Innovation using research and development levies of the horticultural industries. With the significant co-investment from the states and territories and contributions to the Australian Honeybee, or from the Australian Honeybee Industry Council, Grain Producers Australia and the Australian Government. The National Bee Pest Surveillance Program is coordinated by Plant Health Australia and delivered by the states and territories, and that's where I come into play now, delivering the port surveillance work and training and education under the National Bee Pest Surveillance Program for New South Wales. So going through the port of Botany, Kembla and eventually Newcastle once we get bees back into Newcastle, the sentinel hives, and chasing exotic pests and diseases that could be of a threat to the horticulture and honeybee industry. So and my role can involve conducting floral sweeps around the ports and actively looking for a range of exotic pests and diseases that are still out there. I know we've got the portfolio is growing, but there's still a lot of other threats that are out there, and that's where my role comes into play. So that's my story so far in bees. But, so we're actively on the hunt for exotic pests and diseases like the varroa variants. I think there's a fair few, or there is a fair few of varroa variants there. Acute bee paralysis virus. There's a portfolio of them all there of what we're looking for, what I'm actively going out to look for, and that we need to encourage all the registered beekeepers Australia-wide to be actively looking for as well. So one being one of the biggest threats at the moment, I've tried to put it in an order of precedence of what I think would be a priority, would be what Dr. Coops would have covered on earlier today, I'm sure, no doubt, and everyone else. Tropolaelaps mites, or trophy mites as Professor Sam Ramsey likes to call them. You might be wondering why Sam put a varroa mite here. I've just put it there in contrast to give you a scale or an idea of what a trophy mite or Tropolaelaps mite will look like in comparison to a varroa. There's some images there of trophy mites feeding on a larva, and they're native to Asia, where the honeybee evolved to parasitise the brood of the giant honeybee, so Apis dorsata, which I can touch on very shortly. There are 4 species of Tropolaelaps, and 2 of which are known to feed on European honeybees. So not only do they parasitise the bees, they're capable of transmitting viruses as well, like deformed wing virus, very similar to the varroa mite. But another issue or another potential threat is a tracheal mite. That's an exotic pest. They're microscopic. They're quite small, untrained to the human eye. Tracheal mites, they invade the breathing tubes or the trachea of the honeybee, and that's what, hence the name of tracheal mite. They can't be seen to the naked eye, and they're less than 0.2 of a millimetre in size. So these mites crawl down into the trachea and pierce their mouth parts into the lining and draw up the blood or the hemolymph of the honeybee. And they can only be confirmed under a microscope and via an expert identifier in the lab. But the image behind me is a trachea that's been dissected with some mites inside it. So that's a close-up image on the far left. Just another thing that we're looking out for. So another symptom that you may pick up if you did have tracheal mites in your hive would be this K-wing, where the wings are quite splayed out, and also high levels of colony losses. So that could be an indication to get it tested in a lab under a microscope. Another threat is the Asian hornet. So Asian hornets, they're predatory, capturing and killing adult honeybees and brood to feed to their own larvae. Adults are 25 to 35 millimetres in length, and their nests have 5 to 6 layers of like a paper mache-like material in the image to the centre there. It's like a creamy beige or a brown in colour, and with a single entrance hole of about 15 millimetres in diameter. But yeah, this image above my head here is quite confronting with the hornet there having a feast on a honeybee. So yeah, that's why they are on the exotic pests list. So yeah, that's what their nests look like, just to give you some indication of what we're looking for. So virus loads, yep, both varroa mites and Tropolaelaps mites can be carriers and vectors, and other bee species, subspecies can be carriers for viral loads. And one of the worst being deformed wing virus, which hasn't been detected or identified in Australia yet. It's big yet. We've been looking throughout the surveillance work we've been doing, and I've seen symptoms similar to this and had them tested for deformed wing, and they haven't come up. It was just the fact that varroa had taken over the hive that much that the wings hadn't properly developed. It was very similar symptoms to what we're looking at here. And there's plenty of virus loads that could be, viral loads that could be out there. Acute bee paralysis virus is one of them, and slow bee paralysis virus. So slow bee paralysis virus is found throughout Britain, Fiji and Western Samoa. And that's where the front legs become sort of paralysed. So I don't know if Dr. Coops has already covered this today or not, but I just thought I'd mention it and put it out there just to give you an explanation of what threats are there. The large earth bumblebee is another one. So this is an image that I found when I was working down in Tassie, actually. So it's endemic throughout Tasmania, the bumblebee. And I was just blown away at the size of them. You look at it fits in the palm of your hand, the size of a golf ball. They're bloody massive, massive bees. But yeah, that's from the Tocal in the northwest of Tassie, that image. But yeah, there is potential for resource competition and like damage to native flora because of their size when they come in and they hit the plant so hard. But yeah, this is a comparison of the abdomens of a heap of. See if I can move myself across here. Here we go. Your beauty. This is a comparison of the abdomens of some, or Apis dorsata, which is the giant honeybee on the left here, and then European honeybee abdomen in the centre. And then to the right here is Apis cerana, the Asian honeybee. So I just thought I'd give you an indication of the abdomen is one of the big indicators to be looking out for exotic bees. But yeah, the list goes on. Here's another image of dorsata. That's the giant honeybee. So they can carry Tropolaelaps mites and tracheal mites. And they've been known to show signs of deformed wing virus as well. So that's why it's on our radar to be looking for actively. And there's a few others. The red dwarf honeybee, or Apis florea. So at the moment, I'll move myself over here. Your beauty. Apis florea. There's actually they can spread Tropolaelaps mites as well, and also be carriers for other varroa variants like EU varroa. And currently there is detection of Apis florea in Western Australia at this point in time. So it'd be interesting to see what comes of that. They're a much smaller bee. There's a queen there to the right of the left-hand image, and that's what their comb build-up looks like. But they swarm quite often as well. But yeah, another potential threat to industry. So that's what we cover. So there's other endemic pests and diseases that we have. as well that we need to keep an eye out of. I've sort of put this in an order of priority because this has been one of the biggest threats. Excuse me. One of the biggest threats to the bee industry in New South Wales or East Coast Australia at the moment was the small hive beetle and how they take over. This will be an interesting combination with Varroa as we learn to live and manage with Varroa. There's an image there to the left that a lot of beekeepers would be very familiar with, which is a slime out. It's the larval stage of the small hive beetle. As it defecates throughout the hive and eats all the matter throughout the hive and can tarnish it. High levels of yeast. They can detect a hive that's being overwhelmed by small hive beetle. Very, very versatile beetle. It'll be interesting to see what happens as Varroa gets established here as well. American foul brood is another big one. It can spread throughout all bee equipment and movement. It's been a bacterial disease that's remained viable for over 50 years in beekeeping equipment and hiveware and wax and honey products. So it's a notifiable disease under the code, and it needs to be dealt with by euthanizing the hive and burning or treating the hiveware with gamma radiation. So that's a rope test there, that image there of early detection. I've got an image, I believe, of a healthy brood to the left there, as you can see, and then over to the right, there's quite a heavy infestation of AFB with sunken cells. I don't know if you can see the quality of the image there. There's a lot of scale on the old cells here, but it's a terrible disease for honeybees. It's quite endemic. I've been blown away at how much AFB I've found in both recreational and beekeeping commercial enterprises throughout this response that I've been involved in. So yeah, it's a big one on my radar that we want to try and educate industry and recreational beekeepers alike to deal with these issues accordingly. So Nosema is another one, Nosema apis. It's like a dysentery in and around the entrance of the hive. There's another symptom is loss of production and reduced brood production. Worker bees can be crawling around the hive with like swollen and greasy looking abdomens. But yeah, it's another one that we don't want to, or we want to address as best as we can. Chalk brood is another reportable, notifiable disease. It's a common fungal disease, common spread. It can remain viable in wax and honey for several years as well, and it affects the brood stage in the life of the colony. But yeah, there's some images there of little mummified carcasses here to the right, and that's how they get their name, chalk brood, and that's what it looks like when you're looking into the hive. European foul brood, it's just another irregular, it's another concern that's endemic disease throughout Australia at the moment. Irregular brood patterns with a mixture of capped and uncapped cells. You get dead larvae like in twisted positions. There's an image there just behind me of a larvae that's been removed from its cell. So it can be confused with AFB, but it's mostly distinguished between the two diseases by doing that rope test that I just had in the image prior when I was showing AFB. AFB will rope, whereas European won't be as likely to rope at all. But yeah, there's other established pests and diseases like sack brood, greater wax moth, brawler fly, which is endemic to Victoria now, I believe, and but not yet New South, and black queen cell virus. So there's a big portfolio of nasties that are there. If you do detect anything here, contact the plant pest hotline. I'm going to promote that. 1800 081 881. or you can go to the New South Wales DPI homepage under the Biosecurity Food and Safety tab for reporting the notifiable pests and diseases. I can show you that image now. So this is what it looks like on the DPI website. When you get on, you come up here to this Biosecurity Food and Safety tab, and it's just the one at the top there. Follow the prompts for reporting a pest or disease. So that's what we're asking and what we're promoting throughout the state and beyond. And I've got another one for a QR code for sharing reporting your washes as well. So that's if you want to report it's recommended you do your 16-week reports for your washes. That is a QR code that will take you directly to the link as well to report your washes. But yeah, that's me, and I appreciate all your time, and I look forward to getting to know you. That's my story in bees. I'm really interested in getting to know what your story is, and I hope I serve you well in this new role, in this new phase in beekeeping. I never planned to be in this space. I wanted to be out in the field, but here I am, and it's an absolute privilege to be involved in bees. So thank you for your time. I hope you enjoyed the day.
I'd now like to introduce Dr. Chris Anderson, Deputy Chief Plant Protection Officer for New South Wales DPI.
Thanks very much for joining us today. My name is Chris Anderson. I manage the bee biosecurity program in New South Wales. Have also consequently been involved in the varroa mite response. And I'd like to just talk you through a few things today about our bee biosecurity program, and also give you some updates around the most recent version of the varroa mite emergency order, which we expect will now stay in place, establishing some fairly clear guidelines around how to move within New South Wales as a beekeeper. And we have worked very hard to reduce a lot of what was considered to be regulatory burden on beekeepers, and to keep it to an absolute minimum, whilst we still attempt to manage the risk of varroa in New South Wales and prevent it from rapidly spreading. So firstly, just a bit about the New South Wales Bee Biosecurity Program. Under the National Bee Biosecurity Program, we have one funded role, who is Rod Burke. Many of you will know Rod. Rod's been around for a while now. He's very good at what he does, and he's very approachable and available. Whenever you need assistance or advice, Rod is a great person to reach out to, particularly for commercial beekeepers. And the good thing is we have recently been able to fund an additional 2 bee biosecurity officers in New South Wales. One of those I'd like to introduce to you as Sam Jiggins, who has started with us coming from the varroa mite response program. So Sam is now an additional resource for the industry. He's there to provide advice and hands-on support, especially in a post-varroa world that we're now in. And the other bee biosecurity officer, who we're currently finalising recruitment for, will be announced shortly. And both Sam and Rod and the new BBO, their work is coordinated by Megan Power, who sits in the Plant Biosecurity Unit. And between the 3 of the BBOs, they work across the National Bee Biosecurity Program. So this is a national program that ARBIC and Plant Health Australia oversee. And so we see either fully or partly funded bee biosecurity officers in all of the states and territories. And then the National Bee Pest Surveillance Program, which is the program that manages the sentinel hives at the ports of entry across Australia, and is the program that detected varroa mite in June of 2022, leading to the response. And these 3 BBOs will also be playing a very, very important role under this transition to management for varroa as we move away from eradication and to a management phase for varroa mite in Australia. So their email addresses are there on screen for you, Rod and Sam. If you need to take note of those email addresses, please do, and please contact them whenever you need to. Any questions around beekeeping, they will be more than happy to assist you. So one of the key things as we've moved away from eradication to a transition to management program is the creation of 2 new zones. So we have the management zone, which is broken into 2 parts, one around Kempsey and one around Sydney and the Hunter and the Central Coast, and the suppression zone, which is the rest of the state. So these 2 zones replace what used to be called the blue zone, the purple zone, and the red zone. Those management zones are based on the areas that we know have the highest levels of varroa mite detection in the state. And so you can see this heat map here, and this heat map is something that DPI will continue to develop as we roll out this transition to management program in New South Wales. And the goal of this heat map, which is available on our website, is to help you understand where varroa mite has been detected and what its levels are at in those areas. And this is key for you in terms of making decisions about where you might or might not want to move hives to, or if you do want to move hives into one of these areas, the fact that you're going to need to protect those hives by applying treatments and monitoring for varroa mite if you're going to go into one of those heavily infested areas. So what are the rules? What do I have to do as a beekeeper in the suppression zone and in the management zone? Well, for both of those zones, the monitoring requirements are the same. So you need to monitor each apiary for varroa at least every 16 weeks and treat at a threshold within 14 days of the positive test. That threshold is 6 mites or more in a single alcohol wash, soapy water wash, or sugar roll test. So 6 mites or more in one test. That's the threshold, and that applies at this time of the year. Liz Frost will talk more about those thresholds and how they differ. But for the spring, summer and autumn, generally speaking in Australia and under this order, the threshold is 6 mites. That's what you need to be looking out for. If you're at 6 or above, you need to treat. So every time you test an apiary, so the testing, remember, is at the apiary level, you must keep records and report those results to DPI, negative or positive. Doesn't matter, because that's what we're using to inform that heat map, which in turn is informing you and giving you information around where varroa mite is and where it isn't, and what the levels are like across the state. There was a contentious requirement to submit a photograph in previous orders. That has now been removed. So to report the results of your alcohol washing, your sugar roll test, or your soapy water test, you can use the online form, or you can call those results through to the 1-800 number. Now, how do you do this? So how many tests do you need to do in an apiary, and how do you work out whether you're above or below threshold? So you need to test the greater of 10 hives or 10% of hives in that apiary, whichever is the highest number. So here there's 3 examples. There's 20 hives in each one of these apiaries, and in the first example 10 hives have been sampled and no mites have been detected in the test. So for each hive there's 1 test. You only need to do 1, and there's no mites detected in any of those tests. So you've done 10 tests, 1 test on each hive. There, you've met your requirement. You don't need to do anything other than continue to monitor. In the second example, 10 tests have been done. Mites were found in 3 of those tests, but all of those tests came in for this time of year at below threshold. So as for the apiary where you detected no mites, what you need to do is continue to monitor, but you don't need to do a treatment. There's no regulatory requirement to do a treatment in that scenario. But in the third scenario, as you can see, we've tested another 10 in an apiary of 20. However, one of those hives is above threshold. So this time you do need to treat, and you need to treat all 20 hives that are in that particular apiary. The hive that has 8 mites in it is above threshold. Don't get confused, because the hive that has 5 mites in it is below threshold. But if it's 6 and above, it's above threshold and needs treatment. So treating hives, where do you get treatments from? It's a great question. A lot of people are asking it. You can obtain Baverol from DPI, but you can only get it if you're above the threshold. There are limited stocks and they are running out. So you need to call 1-800-084-881 if you want to obtain Baverol, and you will need to show that you've met that threshold. You can also go to retail and wholesale suppliers, and the best I can do is tell you to go and Google it. At the moment, most people will be aware that one of the major retailers has come online and is selling Baverol. They will start selling additional products shortly, as will several other suppliers. So products that we expect to be widely available soon include Baverol, Apistan, Apivar, and Formic Pro. As you can note there, only one of those is an organic treatment. A lot of questions about oxalic acid. There's a lot of work going on to prepare a case for approval and registration with the APVMA. Oxalic acid in beehives needs to be veterinary grade, and that requires manufacturing at certain standards. Industrial grade oxalic acid for cleaning concrete is not suitable to use in beehives, and it is illegal to use in beehives. So despite the fact that some people might think to do that, and we have seen that through the response, that is an illegal use pattern. It's better to wait for the APVMA to approve a veterinary grade product so that we can get access to that and treat our bees with it. What about moving hives within each zone? So we've talked about monitoring and treatment. What about moving? Well, it's pretty simple. To move hives, it's the same as it always has been effectively. Complete a hive movement declaration form when you're moving those hives. And when you do that, it requires you to declare that you have done the Tocal online training and that you're compliant with the monitoring and treatment requirement. Why are we continuing to require this? Well, mostly this is around interstate movement and the requirements of states like Queensland and Victoria and South Australia, that they want to see traceability in New South Wales if they are going to consider reopening their borders for New South Wales-based beekeepers. So we are keeping these requirements in place. Some of the questions we're getting: if I'm above threshold when I do my monitoring, do I need to treat my hives before I can move that apiary? And the answer is no. The monitoring requirement is that you must monitor. If you're above threshold, you must treat within 14 days of that detection. Do you have to treat at the same location that you detected the threshold? No, you don't. You can move that apiary somewhere else and then treat it, so long as the treatment occurs within 14 days of that detection. That's the key thing. Now, moving hives between zones from the surveillance zone to the management zone, not an issue. You can do that, fill out the application form, be compliant, a minimum of 16 monitoring, treating above threshold 14 days. Management zone to the surveillance zone, no, you cannot move colonies into the surveillance zone. If you want to, then you must apply for a permit and your individual case will be assessed. And if there is a way to minimize or mitigate the risk, then the DPI will talk you through that. And it may be possible, but as a blanket rule, the answer to that question is no, and you must apply for a permit. So there is a number of other things that I haven't talked about here, including honey supers. Generally speaking, within each zone, honey supers can be moved around, but there may be a few minor requirements. It's best that you check the DPI website where you can find the most recent copy of the order. That order is now in place and is unlikely to change until such time as we may move to a biosecurity order that is not an emergency order, but it will likely stay in place up until the point where DPI has completed the voluntary euthanasia and disposal of hives that a lot of beekeepers are requesting. And so it's likely to stay in place at least until that work is completed. And the other thing I need to point out and really emphasize to you about those zones is those zones are time limited. They will not be in place forever. The maximum time frame that they will be in place is 12 months. So in particular for the management zone, it's 12 months since it was declared. So it will not be in place for longer than 12 months. The purpose of those zones is simply to slow down the potential rapid spread of mites into the rest of New South Wales. But we do expect that there will be levels of mites building up in other parts of New South Wales. And so as that starts to equilibrate across those zones, it will make sense to remove those restrictions on hives coming out of those areas. But if you are a beekeeper who is in a management zone and you want to move supers out, you can do that. That's not an issue, but you need to comply with the rules around those honey supers. So you can manage apiaries that are out in the suppression zone. You can have your shed in the management zone. There's no issue with that. But you just need to comply with the requirements that are in the order. So please visit the DPI website for more information around the order, where you can find a copy of that order, read it and understand it. And if you need any assistance or support with, particularly with Varroa, but with AFB and other diseases and pests, please don't hesitate to reach out to a bee biosecurity officer. That's what they're there for. And I hope this presentation has been informative and useful. Thank you.
I'm pleased to introduce Bianca Giggins, Australian Honeybee Industry Council.
Hi, my name's Bianca Giggins. I'm the Varroa Coordinator for the Australian Honeybee Industry Council. I'm bringing you a really brief update on the work that's been happening in ARBIC over the last 12 months on behalf of our ARBIC Executive and CEO, Danny Lefebvre. So thanks for having me, Tocal Beekeeper Field Day. A little bit about our structure. I'll just pop out of the way. So we have 10 member bodies which help us decide on the best interests and needs of beekeepers across Australia, made up of different state associations, honey packers and marketers, queen bee breeders and crop pollinators. And they help us to advocate for beekeepers through to some of our stakeholders in industry, which formulate in different ways. Some of them come under the Department of Ag. Some of the collaborations we have are with Plant Health Australia, including our biosecurity network. AgriFutures to support the honeybee panel to facilitate training and different research that happens within the industry. And also connections to our pollination dependent industries as well, which is very important to the support for the honeybee industry. Funding for ARBIC doesn't come through levies. We do not receive any levy funding at all. All of our structures that sit under ARBIC and all the work that we do under our strategic plan are funded by our Friends of ARBIC model, which a donation can be made through our website. We do receive small amounts of money from our memberships in our membership bodies. We also advocate to receive external funding on occasion, but most of it is through the Friends of ARBIC model. So to support the industry to do its job and to make honeybee industry successful in Australia, being able to support ARBIC is a fantastic way of making sure the industry has a bright future. ARBIC is made up of a committee, which includes executive and chair, and subcommittees. So the subcommittees are really important. They're part of every state association has the opportunity to nominate a representative for each of our subcommittees. And it's through that work of the members in those subcommittees meeting throughout the year to formulate the needs of beekeepers on the ground up to the ARBIC executive and the CEO to help us make decisions. And a lot of this correlates to our strategic plan. So the subcommittees sit there with education, resources, biosecurity, queen and importation, imported honey, and recently a levy committee as well. So our activities to date, especially formulated around the Varroa response with Industry Liaison, our representation at the Consultative Committee for Emergency Plant Pests and the NMG for this response in Varroa. We've also been working very hard in the background with the Response Plan version 4 draft, advocating for the best interests of the national honeybee industry through the transition phase that we're currently involved in at the moment in Australia. We've been advocating through projects and funding that we receive. Some of that is through our ARBIC's new branding. For my particular role to assist ARBIC in helping to manage what's happening in the Varroa response. and other funding that we've received for the Bee Pest Blitz campaign, which is coming up in a couple of slides to talk to beekeepers and train them and give them information about how to do alcohol washes and conduct proper monitoring for Varroa in their colonies, and other exotic mites, mind you. So ARBIC's activities also include part of the work that happens in the subcommittees. Our appointments at different other committees around the country help us to formulate good outcomes for our industry at different levels, not only government, but with different boards that affect our industry together. Part of the Varroa response at the moment is that we're in an interim phase where we're transitioning the national Varroa response to a management phase. So we're continuing to work with New South Wales and Victoria in that space daily. You can actually update and review some of our industry updates from ARBIC's website by scanning the QR code or by visiting our website. So it's expected that the transition to management provisions will enable a short-term, nationally cost-shared program that will equip the honeybee industry to be able to deal with Varroa in the future. We promise, we've been working hard in the background, and including some of the work we're doing with the New South Wales DPI and the APVMA to ensure that beekeepers have access to management tools and treatment options. Our most recent update does provide a fair bit of information for beekeepers who are interested in understanding what's been happening in the opportunities for treatments that are actually permitted in Australia for honeybees and Varroa. Our key achievements this year, since our last update to you at the Tocal Beekeepers Field Day last year, is we've run 4 industry Varroa webinars. We've established a honey fighting fund. We've put a bee pest blitz campaign together to assist beekeepers to understand how to manage and monitor for mites and exotic pests. We've got a strategic plan that's been drafted and is in the final stages of being completed and review. We've participated in several state conferences, Senate inquiry for biosecurity, and lots of other events across the country that see the best interests of honeybee business put forward, and we really do advocate at every opportunity that we can to make sure that beekeeping industry is supported and is represented as much as possible. So a little bit about our imported honey fighting fund. We understand that there's current threats to the Australian honeybee industry, and it's never been more important to promote and protect Australian honey, and that ensures our long-term viability. So the Imported Honey Subcommittee have sought advice from some experts around the country and have begun collating that information to finalise what particular tests we'll be doing. We'll be taking randomly off-the-shelf imported honey. We'll be using this as a data-driven DI to advocate for policy reform to government. So if you would be interested, please scan the code or visit our website to find out more about the Imported Honey Fighting Fund, and then that way you can contribute to some of the work that's been happening and a future that really does support the hardworking beekeepers of Australia to have the best price possible for the honey they produce. Another one of our fantastic campaigns that we've been launching through the help of a funding resource from the Australian Federal Department of Ag is the Bee Pest Blitz campaign, where we've collaborated with Plant Health Australia to create this web page that helps beekeepers with resources on how to monitor for exotic mites. And this is going to be an integral part of some beekeepers' lives in the next few years. The resources there are all about alcohol washing at the moment. A video is currently in the process of being provided to this website. You can scan the QR code. Once you're on there, beekeepers can actually click on the jurisdiction that they're working in, and they can report their results to the proper place for their beekeeping state. So have a look at those if you are a commercial beekeeper or a recreational beekeeper. There's some really good information on that site, and it will continue to grow as we develop the webpage going forward. So a lot of our lobbying and advocacy work in ARBIC does go unnoticed. I can assure you that it ties in with all of our strategic plan, and the generous donations that we receive as our friends of ARBIC support us is purely to advocate for industry at all different levels, and you'll notice that some of these points all line up with ARBIC's strategic plan. So imported honey is a major concern for beekeepers in Australia. The levy system is currently having a review, so we're clearly involved in lobbying to make sure that's right. We've been requested to include recreational beekeepers where possible. There's a levy reform bill happening, so we're involved in that. Our biosecurity systems, we're ensuring hopefully that it can be sustainable in a model moving forward as we're dealing with Varroa now, but we need to make sure that our biosecurity systems are fit for purpose. And ARBIC is the national body to be able to lead that. So with our biosecurity subcommittee and all of the participation of our member bodies in our meetings, we do our best to be able to advocate for beekeeping industry across Australia. So that really sums up, and I hope that you've enjoyed that little bit of a snippet in what we do. Please always look for the label and buy Australian honey and support the work of ARBIC where you can. And this helps beekeepers in Australia be profitable and have a bright future. Thank you very much for having me, and I hope the rest of the field day goes well. It's fantastic to be part of the longest running beekeeping field day in Australia, so thank you, Tocal.
Our next presenter is Ray Hull, Executive Councillor for the New South Wales Apiarist Association, giving us an update on what the New South Wales Apiarist Association can do for you.
Yeah, hi, my name's Ray Hull. I'm a beekeeper for 40 odd years, a member of the New South Wales Apiary Association on an executive level. That's your peak body for representation as beekeepers. We make representation to all government departments, forestry, DPI, all that, national parks. We also deal with native land titles and court cases where beekeepers are sort of, their resources have been restricted or whatever. There's a whole ongoing saga with everything, as in the orders being made for Varroa mite incursion and the movement to management. We sort of have a bit of a say in how that works so that the boys can get it all right and everybody can work well together. There's a fair bit of stuff that goes on with training. Tocal run a pretty good training organisation and get involved in a fair bit, which is the way to go. The national parks, we're always sort of lobbying for resources. That's our main problem, or one of the main problems with beekeeping is the resources keep disappearing, whether it be through housing developments or subdivisions or locked up in native titles or national parks. We keep trying to advocate to get more land, more resources opened up for beekeeping. We also sort of stick our beak into pollination and make sure the contracts are fair for beekeepers as well and not all one-sided sort of stuff. But yeah, basically we have a pretty good time doing it. It's sort of the way to go. You've got to be, it's your industry, be part of it, be represented by us. What else do you want to know?
Become a member.
Yeah, become a member and support your industry. Be a part of your future. It's your industry, be a part of it. That's it.
Introducing Lamorna Osborne, Amateur Beekeepers Australia.
My name is Lamorna Osborne and I'm the president of the Amateur Beekeepers Australia, which was started in 1954 by Colonel Pulling to promote the cooperation between home amateur beekeepers, professional beekeepers and scientists to better beekeeping standards in Australia. As of last year, we had over 4,500 members in 35 clubs, including as far north as southeast Queensland and as far west as Alice Springs. This has been a huge swelling in amateur beekeepers numbers since 2009, when we had just over 300 members. The ABA Club supports both individual members and clubs. We provide support for individuals, providing them with a newsletter, up-to-date information generally, and insurance. We provided the biosecurity code hard copy when it came out to individuals, and we also provided the sugar shaker, which was useful in detecting Varroa. We also provided the bee brood uncapping tool for clubs. We also provide grants. That helps clubs to get speakers and pay for their travel and accommodation, or they can use the money to pay for equipment that is helpful to them. We also provide IT backup and a website which is undergoing refurbishment at the moment. We also provided just recently samples of Varroa mite in resin, which will help clubs to let their members see what Varroa looks like for real when they've never seen it before. It has been a year of grief and frustration. The grief, really the mass euthanasia of hives in an effort to eradicate Varroa mite has been palpable and painful for those who may have been participating as volunteers and as victims, and some have lost livelihoods. Now, in the management phase, the ABA wants to provide support to those people, and I have personally, and my club in Illawarra, has offered to supply nucleus hives to those who want to restart once the legislation has been decided on. There have been many frustrations this year regarding incidents in the execution of the eradication plan, too numerous to mention here, but they were heard and listened to, at least by this association. We've asked for a meeting with the Minister for Agriculture and are waiting on a date to be set. On a positive note, 7 members of the ABA went to Chile this year in September, self-funded, to attend the 2-yearly worldwide Apimondia conference. We discussed Varroa management with countries that have been doing it for decades and are still managing to pollinate and to produce honey. We were also surprised to hear that southern Italy and the EU are terrified of their incursion of small hive beetle, which we have managed successfully here in Australia for over 20 years. Over the next year, we're here to provide support and structure for beekeepers at a very reasonable fee of $25 plus 20 insurance if you want it. We are lobbying actively for more active listening from government departments. Yours in beekeeping, and may the force be with you.
Next is our Q&A session. A panel of experts will answer any questions arising from today's presentations.
Okay, welcome everybody to the Q&A for the Tocal Beekeepers Field Day. We've had a few questions come in and we've got our panel of experts here. So we'll get straight into it and answer some of the questions that you've sent in today. Emily, we might start with you. For mite monitoring, where do we sample if we don't have a frame of capped or uncapped brood?
Yeah, great question. So in that case, yeah, maybe you have a queen that is new and just starting to lay, and then there's only maybe a frame of egg in there. That's okay. Just sample from wherever you can find the most brood. At that point, most of the mites are going to be on the adult bees anyway. Or in the case where you have a queen that's maybe winding down or it's queenless, so you have a capped frame, only capped frames of brood, just sample from that capped frame. Again, you're going to have a lot of mites on that that frame or on your adult bees rather than in the brood if you don't have a lot of brood in there. And this is in the circumstance that you have mites in the colonies already. So anybody have anything to add there? Okay. Thank you.
Great. Thanks, Emily. Cooper, one for you. You mentioned in other countries that they weren't able to stop the spread of Varroa between the islands. Do you think there's any lessons or specific lessons that we might be able to apply here in the Australian context to keep Varroa out of, say, WA, Northern Territory and Tassie?
Yes, absolutely. Hopefully we can keep it out of those areas for as long as possible. But obviously for the land mass, if you got any bees moving across the country or between those areas is going to obviously exacerbate the risk. That's very obvious. But I'm sure those states will be doing a lot to try to reduce any bees coming into those states. I think there's a lot of lessons in that. It's the onus is on every beekeeper, and to be really looking out for those mites, for those states to be able to have access to sticky mats and strips, so that if they do have a detection, so they can sort of deploy them pretty quickly to be able to work out how far that mite is sort of spread would be pretty ideal. And also just different mechanisms. I mean, if you look at New Zealand, Varroa mite arrived there in 2000 in the North Island, but the South Island beekeepers there paid a voluntary levy to be able to increase their surveillance. They were able to keep that mite out for about 6 years, so it still wasn't, able to do it in the long run. But yeah, definitely some lessons that can be learned. But I think the emphasis is really the onus is on the beekeeper for everyone to be looking out for it.
Thanks, Cooper. You mentioned that they increased their surveillance in the South Island. Does that mean that they were inspecting every hive that was crossing that border? And do you think that's something we could realistically do in Australia?
I think that beekeepers could do a lot more surveillance themselves. That's for sure. We've all got access to monitoring sort of equipment. It's not difficult to do. It doesn't take a lot of time. So, but in terms of the actual program that was rolled out, there's probably more details in that than we want to have an answer for right now. But basically just increased surveillance through existing protocols and processes through government departments and associations that are working closely with them, and just the beekeepers getting on board with it.
Great, thank you. So I think everyone on the panel would agree that ongoing and continued surveillance of all your hives would be ideal in this situation. All right, Rod, one for you. Do you think it's safe to bring bees back into the management zone?
No. Really, it's time now for us to get back in our beekeeping. Eradication didn't succeed. We now need to learn how to deal with mites, and the best way to do that is to jump in both feet. So get your bees back in, and just start monitoring and just manage your bees, see how they go. You might have some failures, but then you learn from your mistakes, and now's the time to do it while we've got pretty clean mites that don't have viruses on them. So get back into it.
Great, thanks, Rod. One for you, Liz. So someone's sent in a question that they don't want to use any toxic chemicals. Can you cover some options that they can use if they don't want to use chemicals and where they get used?
Yeah, sure thing. So the first thing every beekeeper should start with as a baseline is that monitoring. So knowing when your mite levels are reaching that damaging level. But there's tools that you can implement even before you have, a single mite, such as if you're at a hobby or recreational small scale, you can have ventilated bottom boards. Those are not a full control for your Varroa mites, but it's a little something. Another tool that small scale beekeepers or commercial beekeepers that have the labor and capacity to really intensely manage that brood nest can also use is drone trapping. So putting a frame of drone brood in the colony, which is much more attractive to mites. They'll make a beeline, now we need all these mite puns. They'll make a beeline for that drone brood. They'll get capped and trapped in the drone brood, and then the beekeeper must take the drone brood out before it starts hatching. Otherwise, it'll be a mite explosion and the exact opposite of what you want to do. You can, chunk it in your freezer, dispose of it in a biosecure way, and put another frame of drone brood in as the other one's being capped. So you can just be continually trapping mites in the drone brood. That one is, however, seasonally restricted to when the colony is raising drones. Now, those tactics won't be enough at a certain stage, and you'll have to intervene with some other control. And there's non-synthetic organic treatments that DPI either has registered or is currently looking to register. So right now, our regulatory body, the Australian Pesticide Veterinary Medicines Authority, has approved formic acid and Apigard
To my knowledge, neither of those are yet in the country, but those two will be available at some stage, hopefully soon. But they do have caveats in terms of temperature restrictions. So Apiguard, for example, a thymol-based gel formulation, if it's nice and hot outside, like it is, you'll need to use less of it, and specific amounts, kind of half doses, depending on the size of your colony. So it will pay for all our listeners to go back to the DPI Managing Varroa site and check out some new fact sheets coming up that Kelly Lease has written on how to read your chemical label, which is the law, the safety data sheets relevant to that, so you can understand those temperature restrictions and how to use these chemicals, either synthetic or organic, safely.
Great. Thanks, Liz. And would you recommend a range of strategies?
Definitely. So Varroa essentially needs all the strategies thrown at it. So you start with your non-chemical controls, your cultural and your mechanical controls. And another tool I should have mentioned that beekeepers can kind of take ownership of is selecting for Varroa resistance within domestic stock or through legal pathways of importation where that's already been selected for.
Great. Thanks, Liz. All right, Madeline, over to you. So this question's around nutrition. So how can I know the nutrition of the pollen sources that my colonies are collecting?
Yeah, so that can be a bit of a tricky one because sometimes you have pollen coming in and you either I think the first go-to would be to actually observe what sort of colors can you see that the bees are bringing in? Is it just the one color? Are they bringing in different types of pollen colors? And usually if they are bringing in, we say about 3 different types, that is a good indication that they're getting a variety of pollen sources. And even if some of the pollen sources might be slightly deficient, that they're still able to get that nutrition by having access to a range. But there is also some indications to how good that nutrition source may be. And usually that means that different plant species fall into a plant family or into a group, and they can be of similar nutrition in terms of pollen. So for example, one that comes to mind is the daisy family. So that could be capeweed, fireweed, dandelions. They all are orange pollens. So if it is an orange pollen, for example, that's coming in, there's a very high chance that it is part of that plant family. And with those ones, we, for example, do know that they're all low in protein, they have a high fat content, so they have all some similarities amongst them. But there are also some resources out there, and one of the best resources I can recommend is Honey and Pollen Flora of Southeastern Australia, written by Doug Somerville, the famous Bee Floral Bible. And there's some very good information there about the nutritional qualities of pollens and also when are different tree species flowering. So be very observant in terms of what's flowering in your area. And then hopefully with some resources, you'll be able to get more of an understanding of what sort of nutrition the hives have access to in your specific environment.
Great, Madeline. And I just have a follow-up question on that. So if someone's unfamiliar with the flora in their area and they only see one colour of pollen coming in, should they supplement or should they look in the brood box and have a look at a pollen bank and maybe how much brood they've got?
Well, you definitely need to match the resources with what is happening inside of the hive and what the status of that colony is. So and also linking that back to Varroa in ways, I think the most critical part is to actually manage the periods of time where you are suddenly going through a shortage in nutrition. So you might be experiencing, okay, the bees are bringing in good nutrition, 3 different pollen types. Suddenly some resources switch off. For example, right now where it's getting really hot, ground cover is drying up, the bees are not collecting that variety anymore. And that's where it is important to look into potentially supplementary feeding the hives. But that is also involves around your specific area, what are the bees still having access to. But definitely if resources are dropping off and you're managing hives, specifically if you are depending on on what your goals are in terms of the hives as well, whether you're using them for honey production, pollination services, or just trying to keep them as healthy as you can. Supplementary feeding is definitely a good option and something that everyone should be keeping on top of to manage the nutrition and the health of the hive, specifically also in the context of Varroa.
Great. Thanks, Madeline. That was fantastic. Mark, over to you. So we've got a question about what's the recommended wall thickness for a stingless beehive?
They do vary. Most are around your 25 millimetres to 50 millimetres. They're generally around the same external size. Timber in different thicknesses insulates, better. So majority are around the 50 mil. Materials used can be your hoop pines, radiators. It's important not to use a treated pine or anything like that. We generally don't paint inside a hive, just leave it natural. A lot of cypress is used. My findings are around the 50 mil where I am because it does seem to insulate on, in those kind of colder or warmer conditions.
Great. Thanks, Mark. All right. Back to you, Emily. Got another one for you. Is there a certain time of year when it's more important to monitor for varroa mite, or should we just be monitoring all the time?
Yeah, thanks, Kelly. Short answer to that is it's important to monitor in every season as the population dynamics change in your colonies. But it is really critical in the spring as your bee populations are going up, then mite populations are also likely going up with your bees. So definitely important to be monitoring then, and then also when the when the bee populations are going down into the autumn, it's really critical as well because those mite populations can kind of peak right after your bees hit their peak population, and then once the bees start declining in their population, those mite levels can really get really high in proportion to the amount of bees in your colony. So those are really critical moments, but I think, it's important to monitor consistently throughout the season because those dynamics will change. But another thing I wanted to note is that it's also important to monitor right after you do a treatment. So if you put miticides in your colonies because you've monitored and you're surpassed that threshold, definitely make sure to monitor after your treatment to make sure the treatment was actually effective. So, thanks.
No worries. So how soon after treatment should you monitor? Like what's the sort of time frame for people?
Basically, as soon as possible. I mean, as soon as you take that treatment out, it's probably good to do that check. But as soon as you can, I think is probably the best rec. So.
Great, thank you.
Mm-hmm.
Cooper, another one for you. So in your talk, you talked about resistance to miticides and that being an issue and a major problem for beekeepers in other places. Are there any tips for people as we venture on this Varroa journey about avoiding resistance to miticides?
Yes, absolutely, Kelly. So in every other country where Varroa exists, there's resistance to some different types of miticides to different levels and extents. So basically, you're putting chemicals in a beehive and they're not as effective as you'd hope that they're going to be. So it is a major problem globally, and it's really up to us as individuals and the decisions that we make in our operations, whether we're going to be in that basket case of paying good money for chemicals and putting them in hives and they're not that effective. So it's really up to us as individuals. I don't work for Bunnings, but I love going to Bunnings. But it's not the place to necessarily just go on the weekend to buy chemicals and put them in your beehive. You wouldn't go to the pharmacy to buy, like you'd only go to the pharmacy to buy things if you're going to treat, an issue of your own health. It's the same for your bees. You don't just get random things. These chemicals and miticides have been developed by scientists over many years. There's lots of data and research to support their use for the safety of beehives, the bees themselves, the food products, and also human health safety concerns. So using registered chemicals please, look out for what DPI are doing. They're doing an amazing job registering those ones that are used around the world, and they're fit for purpose. Using less than the recommended dose, that's something that I've seen in other countries as well, just trying to take a shortcut, cutting strips up, putting them across different hives. Yes, they will kill some of the mites, but it doesn't mean that they're going to be as effective as they need to be for those mites treatments to be effective long term. Not rotating chemicals, that's another thing. So, and when we say chemicals, I don't mean the name of the packet. I mean the active chemical class itself, the actual type of active chemical within that treatment. So you can have the same chemical that's in two different names or different products, if that makes sense. So make sure you're checking that out. Not removing strips, that's another one. So the mites are just getting, access to a little bit of chemical, but not enough to knock down the majority of them. So a good tip there is just if you're not already writing on the top lids of your beehives, keep a note. When are those treatments going in? When do they need to come back out? Put it in your logbook as well. So there's some tips trying to reduce resistance issues.
Great. Thanks, Cooper. So what I'm hearing from all of that is read and follow the label directions exactly and don't take shortcuts.
Pretty much.
Great. Thank you. Rod, here's a one about swarms. So do you think it's safe to keep a swarm that has lots of varroa mites in it? And I might add, is it advisable?
Oh, definitely. Like I said before, we're on the journey to learn about managing mites in beehives. So you get mites in your swarm, okay, manage the mites. That's the whole thing. It's not a risk for any beekeeper that already has some experience. It's another challenge, but most of us have already done a lot of research, and we know that using different chemicals will kill mites. And yes, we might not get those chemicals in before some of the mites get into the first brood that goes into the hive. But when those mites hatch a couple of weeks later, if we've got strips in, those mites are going to die too. So we need to start getting experience with mites. So the only way to do that is to jump in the deep end. So don't wrap yourself in cotton wool. Don't panic when you find your first mites. Start learning how to deal with them.
Great. Thanks, Rod. You heard it here first. Rod says, go out and catch yourself a swarm and let's start managing varroa. All right, Liz, one for you. These are about hot and cold. So someone's asked, does freezing kill mites? And on the flip side of that, is there any evidence to support heating as a treatment for varroa?
Good questions. Yeah, the whole spectrum there. Luckily we don't have Arctic mites. They've originally evolved with Apis cerana in Southeast Asia, so they kind of like it hot. They kind of like it broodness, temperature. That's their ideal. So yes, they will die by freezing, but freezing, I guess I'm not sure where that's coming from. Freezing is a good means of ensuring your equipment is free of mite. Say if you've got honey supers that you've harvested the honey from, if those have come from a zone where varroa mite is present, then they could have varroa mite in them. So if you're operating your barrier system, you might think, okay, well, this might be a risk point where mites could be transferred from one hive to another. So you could put those sticky supers, extracted supers in the freezer for a couple days before you put them out to a new yard. And then the question about mite heating. So yes, that has been looked at as a tool, a mechanical tool for killing mites, but it's still, I suppose, not a reliable method of mite control. So there's some tools that have been developed or studied in that area, but it's not at the, I guess, stage that we can recommend it as a reliable control tool.
Great, that makes sense. Thanks, Liz. All right, Madeline, one for you. So with varroa, will the nutritional requirements of my colonies increase or change?
Yeah, good question. Thanks, Kelly. In ways, like the nutritional requirements are not going to change of a colony, but I think again, what is super critical in that, once we're dealing with varroa in the mix, is to manage those periods of times where nutrition might be slightly suboptimal. And that is the times when the bees will be more susceptible to any viruses and be more affected by the damage that Varroa is doing. So those are the periods of times where we have to really be on top of the nutrition of the hives and make sure that they constantly have access to all the nutrients that they need.
Great, thank you. Mark, a last question for you. What can we do for stingless bees on hot days?
Oh, yes, good question. And that does relate to us here, certainly on the mid-north coast, where we get those days. They will handle the day 35 degrees, but it's when we go those consecutive ones as, kind of like heat wave, we're 35, the next day it's 37, and if we're approaching 40 degrees on the third day, they find it very hard to bring that, and high overnight temperatures occur as well. So they find it very hard to keep that, temperature out of the hive. It does penetrate over time. So they're the biggest risk ones. The good thing about these little guys is most of the family is not concerned if you put a screen mesh over the front and bring them into your air conditioning. Not something we can do with Apis mellifera because most of the family would be quite upset with that. Important there, a lot of hives have a little drill hole at the back or the opposing side of the entrance. Occasionally bees will open that up once you've brought them in, so maybe a bit of mesh over that just in case. So yes, you can do that. Damp towels, the wicking method with the bucket of water next to the hive and a damp towel in the bucket over the hive, that can work. So that the thing is we can close them off for a day or two. Generally, a front will come through, things will cool, and we can pop them back outside. Thank you.
Fantastic. Thanks, Mark. One last question for Rod, and it's sort of a double question. So someone's asked, when can we expect the mite bomb to happen in New South Wales? And a follow-on from that someone has had their hives euthanized a year ago. Can they start beekeeping right now?
Yep. So the mite bombs have been building up for a long time. Because we don't have viruses in the varroa mites, the bees are actually able to tolerate a very high number of mites. Now, I don't know if this will work. I'm going to put my little photo up. I don't know. Can you see that? Not very easily, but that's an alcohol wash of 277 mites from 328 bees in a colony. So that is like 80% infestation rate, that's just out of this world. We're only seeing that because we don't have viruses here yet that are impacting the bees. So the mites in the environment are just building up and up and up. And some of these colonies will succumb. They will get stressed. The hive beetle will get them, or a brood disease or something, and there may be robbing. And it's probably happened since before the end of last year, which would probably indicate some of the natural spread that we were seeing. Lots of mites in the environment. Maybe the colonies were still reasonably healthy, but a lot of field bees were taking mites out with them. In this colony, we're able to get photos of 2 mites on the underside of one bee and mites just crawling around on top of bees. So a lot of mites, and so we are already experiencing mite bombs, but the atomic bombs will come when the viruses get here, and then that's going to be terrible because as well as the mites will be viruses with it. So that's what we need to get ready for before all that stuff happens, Armageddon down the track like New Zealand and everywhere else. But as far as you lost your hives a year ago, and I'm sorry to hear it, I know how it feels, yeah, go and get your bees as soon as you can. It's all about learning to manage mites and doing it now while the mites are not as hurtful on the bee colonies. Gives us some margin of error to learn how to use things and in our different seasons, because like I said before, if the viruses get here, we need to be really, really good at controlling mites and looking at treating when those 2 to 3% thresholds are hitting our bees in alcohol washes. So let's learn as much as we can now and then get ready if things change later on.
Fantastic. Thanks, Rod. And I'd just like to say thank you to everyone who presented a talk today and to our expert panel of speakers today on the Q&A. And on behalf of Tocal College and the DPI, I'd like to say thanks for tuning in to the 44th annual Tocal Beekeepers Field Day, Australia's longest running field day. I hope that you've learnt something today, and if you want more information, please visit the DPI website. Enjoy the rest of your weekend.